undergraduate level and more than 15 years at the postgraduate level, he has made remarkable contributions to academic excellence. He has an impressive 25 plus years of research experience spanning microbial biotechnology, food biotechnology, algal biofuels, and nanobiotechnology. He has published over 40 research papers in peer-reviewed journals, successfully guided 4 PhD scholars, and completed multiple funded research projects. His scholarly impact is reflected in a strong citation record and notable research indices. He has been the recipient of prestigious recognitions, including the Fast Track Scheme for Young Scientist Award from the Science and Engineering Research Board, DST, Government of India, the Seed Money for Young Scientist Award from DGST, Government of Karnataka, and has served as a Senior Research Fellow under the Indian Council of Medical Research. With his rich academic experience and outstanding research contributions, we are truly honored to have him chair this session. I respectfully invite Dr. Kia Sanjay sir to kindly preside over the proceedings.
Good afternoon. So, it's my pleasure to introduce Dr. Somshekhar Gaura Patil for this session. Somshekhar Gaura Patil accomplished a scientist and dedicated researcher, especially in the field of plant physiology. Dr. Patil currently serves as a scientist in the division of plant physiology at the Center for Coffee Research Institute, Karnataka, Chikkamagaluru. He earned his PhD in plant physiology from the University of Agricultural Science, Harvard, where he has demonstrated exceptional academic excellence. Of course, receiving gold medals during his undergraduate as well as postgraduate programs. With over 8 years of focused research experience in coffee science, Dr. Patil's work centers on climate change, crop farming, plant growth regulators, and nutritional physiology. His research regularly investigates the physiological response of crops, especially to economic stresses, and he is actively engaged in identifying and characterizing stress-tolerant coffee genotypes to support sustainable agricultural practice. Dr. Patil has made a significant scholarly contribution, having published more than 65 research papers in a high-impact international journal and chapters. In recognition of his scientific achievement, he has been honored with Junior Scientist of the Year Award and the World Sustainable Agricultural Development Award. Though his dedication to research and innovation, Dr. Patil continues to contribute meaningfully to agricultural science, climate resilient crop development, and sustainable farming systems. So it is indeed a privilege to have such a distinguished scientist among us today. So he is going to talk about climate change and its impact on coffee production as well as its quality. What is it?
Thank you. Thank you, Miss. Thank you. Thank you. Check. Can I get a point? Yeah. Let's project the slide. Pointer. Slide mode. Yeah. Thank you. Here it is. Thank you. Yeah.
Change
Very good afternoon to all of you. Thanks for the introduction, sir. And it will be a very difficult for me to present after the lunch hours, because after the lunch hours, it is a sleeping hours to all of us, including me also. So I suggest the organizers, whenever you conduct international conferences or the seminars, you can keep a coffee. You can keep a coffee and tell them to drink the, because it will stimulate your brains and all. Thank you. So my presentation is on one of the international beverages, that is coffee, and how it is affected by the climate and what are the intricacies of the coffee quality I am going to cover. So to snippet, I am going to cover origin of the coffee, then climate change aspects. If you look at the scenarios in the world as well as Indian scenarios, how we are experiencing changing climate on the coffee, and also quality aspects, and what are the production, what are the technologies we are adopting and suggesting our growers to mitigate the impact of climate change. So let's begin. How many of you are knowing the coffee? Raise your hand. How many of you are knowing the coffee? Because you are sleeping, that's why I am telling you this exercise. Raise your hands first. Yes. So you are knowing the coffee. How many of you are drinking the coffee? Very good. Very good. Very nice. Because most of the South Indians, we will prefer to have a coffee, and North Indians, they are preferring the tea as an international beverage, because our day starts with drinking one cup of coffee, right? So that will stimulate your brains. So that will make you active throughout the day. Okay? So the origin of the coffee is like that. It was originated in Ethiopian highlands in the year, say 9th century. So it was discovered by the Keledi, he is a shepherd. And when he was rearing these goats in the Ethiopian forest, some of the goats got energized. Okay, from then the coffee came to the world. Okay, from Ethiopian highlands, it has come to the, what do you call it? Yemen in the year 15th century, and 17th century it has crossed the Arab world, and it has come to the, again, Europe. So from that Europe, it has come to the India. So do you know who has brought this coffee to India? So it was brought by the Baba Budan. Baba Budan, when he went to the Yemen, he brought 7 golden seeds from the Yemen. He sown these golden seeds in the Chandradrona Parvata. Chandradrona Parvata, now we are calling it as in Baba Budangiri. Baba Budangiri, which is present in Chikmagalur districts of Karnataka states, and it is the one of the highest peak in the Karnataka. So whenever you come to the Chikmagalur, you can visit our place. Okay, from 16th century he brought to the India, and 18th century the Britishers started cultivating this coffee. So 1870 it has become commercialization, and 1925 the one of the experimental station they have established at Chikmagalur, that is called the Mysore experimental stations, and from the that was established in the year 1925 and renamed as 1946. That is called the Central Coffee Research, where I am working present now. So if you look at the world scenarios, coffee, it is grown in 18 developing countries, and in the developing countries we are consuming 30%, and 70% will be consumed in the developed countries. It means that we are growing coffee in the tropical areas and consumed in the temperate areas. And if you look at the world coffee industry engages, this is 100 million people. There are 100 million people that are engaged in the coffee cultivations. And we are growing the coffee in 7 major producers like Brazil, Vietnam, Colombia, Indonesia, India and Honduras. And other countries includes 21% shares. If you look at the Indian shares, we are the 7th largest producers. We are the 7th largest producers now, and it will change 6th and 7th like that. So fifth largest exporters, India is the fifth largest exporters, and area under coffee is 4.80 lakh hectares and production is 3.74 metric tons. And we are exporting the coffee to majorly to USA and Europe, and 2 million people in India, they are engaged in the coffee cultivations. In India, if you look at the Karnataka is the major coffee growing belt, where Chikmagalur, Hassan and Kodagu shares topmost, and followed by the Kerala, Tamil Nadu, and also started cultivating in the non-traditional areas like Assam and Orissa. And also in northeast regions, 7 sister states, they are also cultivating this coffee. Okay, that's why we are the second largest exporters and second largest traded commodity among the agriculture. That's why it is called as a brown gold. So whenever you sip a cup of coffee, you should understand that you are also a part of the global industry. So how we are cultivating coffee? This is the uniqueness of our coffee. We are growing under the shade. We are growing coffee under the shades, and it is hand-picked and it is sun-dried. This is the uniqueness, where in other countries, the coffee is cultivated under the open cultivations, and they are harvesting coffee through mechanizations, and they are sun-drying through solar tunnels and all. But we are not doing like that. We are growing coffee under the shades, we are hand-picked the coffees, and we are sun-drying. We are also having lot many things in our coffee estates. We have the floras and faunas, and also we are having the richest flora and fauna in our coffee estates. So mainly we are cultivating 2 coffee species. One is the Arabica and another one is the Robusta. So Arabica, it is an tetraploid, and Robusta is an diploid species. Arabica, if you look at, it is sensitive to the pest and diseases. Whenever there is a change in the climate, we will see the effect on the coffee plants. That is especially the Arabica. Whereas Robusta is tolerant to the drought and other environmental variations. So this is one of the impact after the establishment of our Central Coffee Research Institute. So the initial times the production was 10 metric tons. If you look at during 24-25, we reached 3.74 metric tons. It means that after the establishment of our Central Coffee Research Institute, we have taken our coffee to the highest, that is 3.48 metric tons. That's why we are the 6th largest exporter in the world. And this is where I am working. This is called the Central Coffee Research Institute. It was established in the year 1925. And last year, December 23, we have celebrated a centenary of our institute. So this is a pride for our nation also. So we are doing a research on plant breeding and genetics, and also the disease and pest management, and also the coffee quality. And among within our CCR, we have 5 regional stations. They are RCS Chundal, which is present in Kerala state, RCS Thandigudi in Tamil Nadu, CRS Chattali, that is nearby your Mysore only, that is Madikeri, and Arvinagar, this comes under the Andhra Pradesh, and RCRS Deepu, which comes under the Deepu. So we are facing the impacts. Now coming to the cultivation aspects for now the impacts of this climate. What we are seeing, we are changing the climates. What is that? We are observing high carbon dioxide concentrations. That is the culprit. Okay, whenever the high carbon dioxide concentration it is there, it increases the temperatures, it traps the temperatures. Okay, warmer temperature, declining glaciers, and increased frequencies of droughts and floods, and cold days and nights, and hot days and nights, and heat waves are observing. So if you look at the predicted climate change, if you look at the carbon dioxide concentration, 2025 we reached, that's a 405 to 450 ppm. Now we are observing 430 ppm. We reached already, and by 2050 we will reach the 540, and 2100 we will reach the 970. And temperature also 4.1 to 1.3 degrees already we reached, and sea level has raised to 3.5 centimeters. This is the predicted climate. And if you look at the state-wise, what are the changes we are saying because of the climate change? If you look at the Madhya Pradesh, that is soybean bowl, so it is varying trends we are observing, and rainfall if you look at declining trend. Okay, so if you look at the Gujarat, there is an increase in Gujarat, Chandigarh, Maharashtra, including the Karnataka, Orissa, Andhra Pradesh, Kerala, there is an increase in the temperature we are observing and also the rainfall patterns. If you look at the Karnataka itself, this is our place where coffee is grown. We are observing temperature in our Western Ghats because coffee is, we are growing in Western Ghats and the Eastern Ghats. In the Western Ghats and the Eastern Ghats also, we are observing rising the temperatures during extreme summer times as well as the rainfall during the summer, during the monsoon times. We are frequently and heavy downpour of the rainfall we are observing in our coffee estates. So, why coffee likes cozy? It is because of the environmental conditions. We are cultivating, as I said, Arabica and the Robusta will be cultivated in India. The temperature which is required for the Arabica coffee is 18 to 24 degrees Celsius. If you look at that one, Arabica requires 18 to 24 degrees Celsius and rainfall 1500 to 2000. If you look at the Robusta, temperature is required 24 to 28 degrees Celsius and rainfall more than the 2000 mm. Whether we are observing this kind of optimum temperature nowadays, it's no. The temperature has crossed 35 degrees Celsius in the Western Ghats of India. Western Ghats of India, 35 degrees Celsius. As I said, the optimum temperature 18 to 24. Whether it is survives? No, not at all. There is a change in the metabolic processes in the coffee plants. Okay, whenever 1 degree Celsius increase in the temperature, there will be a reduction of the photosynthesis by 10%. If the temperature increases 1 degree Celsius, there will be a reduction of photosynthesis by 10%. If you convert that to productions, there will be 15% of the productions will be declined. 15% of the productions will be declined because of the increase in the temperatures. That's why optimum temperature we have to maintain in the coffee plantations. So, there are temperatures, droughts and winds. They are the main culprit. So, whenever there is a temperature increases, the metabolic rates of the plants will going to change and it will impact on the yield and quality of the coffee. If you look at the drought and the rains, whenever the drought is there, it will impact on the flowering patterns. We are going to see some of the abnormalities in the coffee plants. Certainly, if you notice these abnormalities in the coffee plants, definitely you will observe at the final, that is the yield. Yield will be declined. That's why whenever the temperature increases, this kind of pest and diseases or behavior also is going to change whenever the temperature increases. And also because of the wind, there is an, what we can call it as high wind we are going to observe during the flowering times. Whenever there is an high winds, the flowers will drop off and you will not get any yield at all. So this is all the problems we are going to notice in our coffee plantations. If you look at the other countries like Brazil, Colombia, Costa Rica, Vietnam, India and all. This is last year photo where Brazil stands first in the productions and stands first in the export. What they are facing because of the climates, they are observing the frost. They are observing the frost. And Vietnam also, there is no water for the irrigation. There is no water for the irrigations. If you look at the Ethiopian, again there is a drought. If you look at the Uganda, again they are irrigating through pipes and all because they are not getting water for the irrigations. So this is the last year's June 30th they have observed, it is in the Brazil, 30th they have observed the frost and 20th severe frost they have observed. Okay, July 30th again the moderate they have observed, that's why the yield of the Brazil has come down. Therefore, we are getting a premium price for our coffee because this kind of problems we are not at all observing in our Indian coffee condition. Okay, so this is how the temperature, how the rainfall is changing in our Western Ghats. We have detailed and trend line analysis. It shows that the rainfall, if you look at every trend, because summer, rainy and the winter period, if you look at, there is a change in the rainfall patterns. Every window, if you look at, we are observing high rainfall. We are observing high rainfall and the rainfall which was increased is 132.72 mm per year. 132.72 mm. If you look at the temperature was increased to 0.8 degrees Celsius. 0.8 degrees Celsius. If there is a 1 degree Celsius, as I said, it is reducing your photosynthesis. Okay, now we are observing the 0.8 degrees Celsius. This is the maximum temperatures. If you look at the minimum temperatures, 0.9 degrees Celsius. Okay, so now you got it. What's happening in the coffee plantations? One is the rainfall is increasing. Second thing is maximum temperature is increasing and the minimum temperature is also increasing. So temperature is most important for all of us during the flowering times. Okay, so if you look at this 2024 in CCRI we have received a blossom shower during January 37.5 mm we have received, and we have not received till April month. We are telling that coffee needs a stress period. Coffee needs a stress period for the proper flowering. If you give the stress, you will get a blossom at one time. Because we are harvesting the coffee during the month of January. So January, if you harvest, February and the March period is the stress period for all of us. When you give the stress period, again if you give the blossom irrigations, definitely you will get a good blossom. If you not give the irrigations during that time, as I said, after the 45 days, definitely you are going to observe the floral abnormalities. That will impact on the final production of our coffee. Okay, this is the rainfall trend. If you look at, as I said, every year there is an increase in the rainfall. Okay, so if you look at the monsoon times, southwest monsoon and the northeast monsoon, two monsoons we are usually receiving. The southwest monsoons, it has to start by June 2nd fortnight. June 2nd fortnight, but we are not receiving a rain. During second fortnight of the June. It has postponed to the July month. How we are receiving the rain? There is a torrential rains. In a day you are going to get 4 to 5 inches of rain. Because coffee, it has to pass these berries to a 7 stages. We are telling that pinnacle stage, rapid swelling stage, suspended and slow growth, endosperm filling stage, slow growth, again the ripening and the harvesting time. These are the 7 stages. This June and July month, it coincides with our endosperm filling stage. Because 60% of the bean will be filled during that time. When you receive the rain during that times, you are not going to get a proper bean. You are not going to get a proper flavor. You are not going to get a proper aroma. Because bean will not be filled properly with the carbohydrate and sugars. So this kind of problems we are observing, especially during the month of the monsoons. It means that every year there is a postponement of the rainfalls, and every year we are observing a high rainfall. Okay, let's see this climate change on the coffee. Untimely and ill-distributed rain, especially the reddening blossoms we are going to see, and we will be seeing on the floral abnormalities, and also atmospheric temperature. If the temperature increases, it will not fill properly, and low shades. Again, if we are telling that optimum shade is very much required for the coffee plant. If you maintain the low shade, so again there is a hastening of the ripening, so you will not get proper flavor and the aroma of the coffee. And if you look at the increase in the temperature and the rainfall, certainly I said the photosynthesis is going to reduce, and there will be the skin which is present outside the beans, it will stick to the beans. It will stick to the beans, and whenever you process the coffee, so you will get bit cuts and the bits. Okay, again there is an increase in the pest and diseases, again prolonged drought we are going to observe. If you look at the rainfall, as I said, the heavy rainfall during the endosperm filling stage will not get a proper quality of the coffee. So this is the climatic vagaries what we are observing. So both Arabica and the Robusta are negatively affected by the climate change. Arabica shows lower yields and worse quality due to higher mean temperature, and Robusta is affected by weather events. So these are the problems. As I said, whenever there is an increase in the temperature, these are the abnormalities we are going to see. This is padding, star flowers, pinking, and the snake mouth flowers. We are going to observe the flowering during the month of March and April months in Arabica and the Robusta coffee. If there is an increase in the temperatures and if you're not having the proper soil moistures, again we are going to see this padding and star flowers in Arabica. In pinking and snake mouth flowers, we are going to observe in case of Robusta coffee. And also during the monsoon times, if there is a heavy downpour of the monsoons, we are going to observe these diseases. We are going to observe this kind of diseases. This is called the black rot diseases, black rot and the stalk rot diseases, which will impact on the Arabica and the Robusta coffee because of the heavy monsoons. And also the berry drop. Nearly 50% of the berries will be dropped during the month of July and August if you receive a heavy rain. Okay, and also this flare up of the leaf rust we are going to observe, and the white stem borer that is mainly attacking on the Arabica coffee. Whenever there is an increase in the temperature, definitely we are going to see this white stem borer. These kind of the diseases and the pests and as well as abnormalities we are going to observe because of the climate change. So what do we have to do? We have to reduce the temperature now. We have to reduce the temperature. So we have to reduce the temperature by 1.2 degrees Celsius. 1.2 degrees Celsius. So how we have to reduce? That is in our hand. As I said, the uniqueness of our coffee. What is it? What it is? We are growing under the shade. We are growing under the shade. Even the ACs are put on here, still I am sweating here, and some of you are also sweating. This is called the climate change. Okay, still ACs are on here. Because this kind of heat we are seeing in our plantations. As I said, 1 degree Celsius increase in the temperature, there is a reduction of the photosynthesis and it will impact on the final yield. That's why shade is very important. If you grow the shade, if you grow the trees in the coffee plantations, there is a reduction of the temperature by 3 degrees Celsius. 3 degrees Celsius, we can bring down the temperature. If you open up the coffee plantations, it will impact on your photosynthesis. That's why the coffee should be cultivated under the shade. Should be cultivated under the shade. Okay, then by developing the varieties. What kind of varieties? That is the drought tolerant varieties we are developing, and we are telling the planters to grow this kind of drought tolerant cultivars. And also the soil conservation moistures, that's very important during the rainfall. So these are some of the drought tolerant cultivars we are suggesting growers. The Arabica and the Robusta, already we have released 13 varieties, and during our centenary celebrations, we have released 2 more varieties. They are the leaf rust as well as the white stem borer tolerant varieties. Okay, in Robusta, these are the drought tolerant cultivars. If you not cultivate these things, definitely you are going to observe these kind of abnormalities in the coffee plantations. Okay, it will impact on your whole coffee plants. So this is a white stem borer tolerant cultivar, 4595. So next one is the, it's okay, no, thank you. So first one is the drought tolerant cultivars, second thing is the shade. As I said, if you grow the trees in the coffee plantations, so as I said, we are going to reduce the temperature by 3 degrees Celsius. And in our coffee plantations, are telling 50 to 60% of shade should be there. 50 to 60% of shade should be there in Arabica, and in Robusta, there should be 40 to 50% and 30 to 40% in irrigated coffee plantations. If they maintain a less shade, definitely they are going to observe the abnormalities. As I said, the padding and the star flowers in Arabica. And in Robusta also, if they maintain a less shade, they are going to observe the snake mouth flowers and the pinking types. So, they should have 40 to 60 trees per acre. This is a recommendation, and 20 to 30 trees in case of the Robusta. And this is how we are maintaining the shades. It's from the lux meters. If they have 55 to 70,000 lux, means it is meeting the 50 to 60% of the light. And Robusta 75 to 80,000 lux, we are telling the growers to maintain it, and 80 to 90% in the irrigated coffee plantation, so that it will maintain 40 to 50 and 30 to 40% of the shades. So, if you are not cultivating coffee under the shade systems, definitely you are going to observe this kind of wilting symptoms. If you look at the relative water content, it means that the leaf water, the water which is present in the leaf, this is open cultivation and this is the shaded. If you look at the water content, that is 45.8%. 45.8% without having the shade trees. If you cultivate the coffee under the shades, if you look at 82.4%. 82.4% of the water will be present if you cultivate the coffee under the shade. So, we are having a diversification also. We are growing coffee along with the pepper, areca nut, banana and other fruit crops also. Okay, so that the environment in the atmosphere, in the coffee plantation will be maintained properly. These are the some of the tree species we are recommending to our coffee growers. And also we are maintaining our flora and the fauna. If you look at, we have 23 different species of the birds in our coffee plantations. 23 different species of the birds and 10 different species of the snakes. 10 different species of the snakes, and also the floras and faunas will be maintained in our coffee plantations. So, as I said, if you grow the trees, there will be a reduction of the temperatures by 3 degrees Celsius, and the leaf temperature will be reduced by 4 degrees Celsius. So, this is the adaptation mitigation which is very much required for the coffee plantations. So, one is the water harvesting structures, because as I said, every year we are observing the high rainfall, so that we have to capture these waters. We have to capture these waters and conserve in the ponds. So, whenever the plants get a stress, you can utilize this water for the irrigations, especially during the summer times. And also, whatever the leaf litters, if you look at the shade trees, in our shade trees, because of the shade trees, so there will be defoliations. How much the defoliation? You can't imagine. There will be 11.73 metric tons of the leaf litter which will be formed per year, per hectare. 11.73 leaf litters. From that, 50% of our nutrients will be met. If you look at because of the shade trees, there will be carbon stocks, carbon sequestration, whether you heard or not. The carbon sequestrations, there will be 353 megagrams per hectare of the carbon stocks which will be present in our coffee plantation because of the shade trees itself. And we are saying that our coffee is carbon negative. Our coffee is the carbon negative. That's why you are meeting our EUDR regulations, because most of our coffee we are exporting to the European countries. They have brought out some of the regulation, that is European Union deforestation legislation. It tells us that your coffee should be in the forest. Your coffee should be in the forest. Then only they will buy the products. So we are telling that our coffee is carbon neutral. So again, the coffee quality. So we have a region specific coffee qualities, and we are having the Arabica, Baba Budangiri Arabica is there, Chikmagalur Arabica is there, Arku Valley, Wayanad Robusta coffees. These are the specialty coffees. Because of growing coffee under the shades, we are achieving all these. These are the regional logos with the specifications. And significance of our coffee sector is we are conserving our ecology, we are giving employment to the livelihoods, and tribal growers are getting benefit out from our coffee, and we are what we can tell is a rejuvenation of green cover in our Eastern Ghats and the Western Ghats. Okay, this is all about my topic. As I said, the temperature is 1 and the rainfall is 1. If you mitigate these temperatures, if you control this temperature by growing shade, definitely we can achieve the coffee production to the 7 lakh metric tons. So this is our Central Coffee Research Institute. Whenever you come towards Sringeri to visit the Sharadamba temples, you can come to our research station also. Thank you one and all. Thanks for the organizers for giving me an opportunity. So if you have any questions, please ask. Yeah, please.
Sir, other than coffee, we can get any other useful byproduct from the coffee bean.
So definitely. From the coffee, you will get the biochar. Second, you will get the just now I spoke with one of the faculty here. You will get the bricks. After the processings, you will get the lignin. Okay, alkaloids can be extracted, and also the phenols can be extracted from our coffee. These are the byproducts we are going to get from our coffee. Yes. Thank you.
Anybody else?
Is the center open for public sector?
Yes, ma'am. Anybody can use it because this is a public sector.
Here is one more question.
Yeah, please, sir.
Hello, sir.
Yeah, hello.
So, in what way photosynthesis can affect the taste and quality of coffee?
So, if you look at the temperatures during the ripening times, when the temperature increases during the ripening time, it hastens the ripens. Okay, so there will not be a proper filling of these sugars to the beans. Proper filling of the sugars, because it will fasten the sugar accumulations. Some of the sugars which will be present in the leaves itself. Improper filling of these sugars will happen. That's why we will see the impact of these temperatures on the coffee.
Any other questions? Okay, if not, thank you, Dr. Patil. That's a wonderful presentation.
Thank you, sir.
Especially on global coffee facts, climate change impact on coffee, especially rainfall and temperature effect, and how to develop different varieties and adaptation and mitigation process so that we can increase the production process.
Thank you.
So, on behalf of organizing committee, I thank you for your presentations.
Thank you, sir. Thank you. Thank you very much. Thank you. Thank you. Yes.
I thank Dr. K.R. Sanjay sir for chairing this session. Thank you, sir.
Thank you, sir.
an invited talk from Dr. Bettadaya B.K. Sir, Senior Principal Scientist, Plantation Product Spices and Flavor Technology Department, CSIR Central Food Technological Research Institute, Mysore. I request you to kindly take your seat in the dais. We welcome you, sir. It is my immense pleasure to welcome Dr. B.K. Swami sir, an accomplished academician and researcher in the field of chemistry. He completed his BSc, MSc in organic chemistry and PhD in polymer science and technology from the University of Mysore. I welcome you, sir. With over 35 years of teaching experience, Dr. Swami sir has served as a lecturer in various JSS institutions since 1991 and is currently working as an associate professor at JSS College of Arts, Commerce and Science. He also brings 26 years of rich research experience, having published 18 international research papers, presented numerous conference papers, chaired sessions and delivered keynote addresses at national and international forums. He has successfully completed UGC-funded research projects and has been recognized with awards including the Best Paper Presentation Award. Beyond teaching and research, he has contributed significantly to academic leadership, serving as principal, NCC and NSS officer, chairperson of BOS and BOE, and organizing multiple workshops, conferences and seminars. We are honored to have such a distinguished scholar with us today. He will now be introducing our esteemed speaker, Dr. Bettadaya B.K. sir. Please join me in warmly welcoming Dr. Kinnara Swami sir. Welcome you, sir.
Good afternoon, everyone. It's my privilege to introduce Dr. B.K. Bettadaya, Senior Principal Scientist at CSIR CFTRI Mysore. Dr. Bettadaya obtained his BSc, MSc and PhD from University of Mysore. A distinguished academician, he is a gold medalist from organic chemistry and platinum jubilee gold medalist from University of Mysore. Following his doctoral studies, he pursued postdoctoral research at CNRS, University of Paris, France. Subsequently, held key R&D positions in reputed industries, namely Sigma Aldrich. He joined CSIR CFTRI in 2010 and he has steadily risen to the position of Senior Principal Scientist. His research expertise spans food science and technology, flavors from spices, marine substrate, etc. He has published 50 research papers in high-impact international journals, held 2 patents and successfully completed 15 research projects, including mission mode and industry-sponsored project. He has guided 7 PhDs and currently supervising several doctoral candidates and has mentioned more than 20 master dissertations. Okay, he oversees PhD activities more than 250 research scholars. He is also actively involved in academic governance as a member of Board of Studies and serves as multiple institutional committees. Dr. Bettadaya has delivered innovative lectures at international conferences, received Best Paper Publication Award from CSIR, CFTRI, and continued to contribute to national standard as a member of BIS Committee in Spice, Herbs, and Condiments. With his rich blend of academic experience, industrial R&D leadership, and impactful research in food chemistry and neuroscience, Dr. Venkataya stands a distinguished scientist contributing significantly to sustainable food and flavor. With this, I invite Dr. Venkataya sir to deliver his talk. It's over to you, sir. Thank you.
Thank you. Sorry. Thank you. A very good afternoon, everyone. Am I audible to you? Last person. Am I audible? A very good afternoon, everyone present here. And I'm in this my primary pranams to the His Holiness Jagat Guru Sivratri Deshikindra Mahaswamiji and also the organizers of this conference. The organizing chair, Professor Mallu, and all the organizing team who have been asking me to present a talk in this conference, which is aptly or correctly coined the bridging chemistry and the food for the sustainable growth of the society, which is being, the theme of central theme of the conference. I would like to add on to this the subject with the introduction of the spice chemistry and how a sustainable society can be looked in from the understanding the chemistry, not only because of because spice provide the color and the flavor to the food. Okay, behind this one, understanding chemistry, whether we have anything for the sustainable society is the talk of my subject. And many of you are familiar with the palatial building which is shown here, which is a Central Food Technological Research Institute, which is completed 75 years service into the nation. And this is the generous gift from the Maharaj of Mysore for the conducting the food research in this country. We come under the organization called Council of Scientific and Industrial Research. We have all the many research institutes spread across the country. And moving on to the subject matter of spice, everyone knows that spice is our heritage. So India produces a huge quantity of spices, nearly 15.3 million metric tons every year, and we are also the biggest consumer as well, either in the form of the primary processed product or semi-processed product. And also the revenue which you get from the spice export is close to around 40,000 crores. And as we know that the Silk Route is being established between connecting the East and the West, similarly the spice trade or the spice route is also connected between East and West because of the spice, which is also being reflected in one of these the recent movies, Kantaran Chapter 1. I am audible to you? Similarly, the spice trade is being one of the oldest trade practice which is being started, and we have been also looted and then by the exploited for this the treasure trove which is present in India. And this is the export of Indian spices. This is the extracted from the spices board. And if you look at the compounded annual growth rate, which is about 7%, and in terms of the value, it is over the 10%. And Indian spices are being valued all over the world, and we export the spices to every nook and corner of the world, and which gain you the lot of revenue. And if you look at the export commodity wise, chili takes the top priority, and followed by the cumin. And additionally, we have the commodity called spice oils and oleoresins. The spice oils and oleoresins are the value-added products which have been extracted from the dry spice by hydrodistillation method to get the oil, or you can get an extract into a solvent and concentrate a resinoid form of the spice which is called a oleoresin. these spice value-added products are being used and exported to the various other countries because they have the good shelf life and the small quantities are sufficient enough to bring a valuable color and then flavor to the food, and also so it serves a, the logistic supports in terms of the quantity which is being very less compared to the spice alone. Similarly, we have various other commodities which is also being exported from India, and these are these figures. As I already mentioned, the spice provide you a value addition into the food in terms of the color and the flavor, which increases the palatability, which we increase love to have the food more when the taste is good, right? Similarly, if that is the only a case, we would have not been interested to go further detail and to understanding on the other aspects of the spices, but it also give you the goodness of the bioactivity, which is due to the various chemical components which have been composed in the essential oil components as well as in the oleoresin. These are being by the monoterpenes or the alcohols or the aldehyde ketone ester phenolic compounds as well. Additionally, they are also having the medicinal property which is being utilized in various balms or decongestant and pain relief the formulations. Additionally, they have also the perfumery applications, so that the product, consumer product, the variations could also be seen in various types of these type of products. Yeah, when you look at the spices, you have various the concentrations of oils you could obtain, that is steam distilled oil, so which varies between 0.4 to maximum 6%, except for another 2 cases. But the various parts of the plants which use is for the spice extractions or spice utility are like root, seeds, stem, leaves, etc. And also now we are able to see that the essential oil components, a single organic compound is mainly responsible for the intended activity or intended application, such as like one-edicineal, eugenol, jasmine, vanillin, all these things are the single organic compound which are present more than 60 to 70% in the oil alone. Little bit understanding on the chemistry aspect of these spices before we go for a in detail understanding. I would like to draw your attention on looking at the primary 3 molecules, which is methanol, methyl mercaptan, methylamine. They have been differentiated by the presence or a content of the heteroatom alone. If you look at this property, you see that methanol is at lower concentration. If you take into body, it is toxic, whereas methyl mercaptan is a very obnoxious smell compound, whereas the methylamine is very unpleasant odor. Just variation in the heteroatom brings in you lot of value in terms of the perception in the aromatic strength of these molecules. And also if you look at the components like the camphoraces, like a camphor and 1,4-dichlorobenzene, which are having the different structures but have the similar odor. Similarly, we can vary upon the introduction of various the functional groups or small variation in the structure of these molecules. For example, if you take a musk, say a musk odorant, suppose if you replace with the nitrogen, it will be less intensive. If you introduce a methyl group next to the oxygen, it will be odorless. Whereas if you introduce a methyl group, 1 carbon atom after the oxygen, there will be increase in the aromatic strength. Similarly, if you see that the vanillin, which is an aldehyde which extracts from the vanilla, which is being, if you say the intensity is 1, the just change in methoxy to ethoxy group will bring you the value in terms of the more aromatic strength or flavor strength about 3 times. That means a single small transformation of the molecule, native molecule, will bring you in larger perception in the odor quality and also increasing the aromatic strength. This has been also very much applicable in terms of the stereochemical structures as well. Suppose variation in the small, the enantiomers, stereoisomers, we will also see that in case of the limonene, which is a C10 carbon system, monoterpene, which is having a strength of, which is a orange odor, suppose the chirality is a 4R plus, or if it is a 4S minus, it will be a turpentine odor. The odor itself will get changed based on the stereochemistry which is available. There are quite big number of such the odor perception based on the stereoisomeric nature we can give. So
Very good example is like a menthol, which is a monoterpene hydrocarbon, which is having the alcohol at the third position, and it has a 3 stereo centers. 3 stereo centers are denoted by 1R, 2S and 5R. There may be, how many the stereoisomers are possible for this molecule? Any chemistry students can say? Stereoisomers. 3 chiral centers. No idea. 4 it is 2 to the power of n. If you say, it will be having 8 stereoisomers. All the 8 stereoisomers are not equally important in terms of execution of the cooling nature of the menthol, except this molecule, which is 1R, 2S and 5R menthol, which is defined by the cooling nature of this molecule from the Mentha piperetica or Mentha canadensis, Mentha auransis. Various species have been, utilized to extract the menthol from the menthol. In this case, chirality, odor perception and flavor activity has been very much, very much interested, and India is the leading producer of natural menthol from the mentha. The obvious reason is due to the introduction of the new variety by the Central Institute of Aromatic and Medicinal Plants, our sister laboratory from the Lucknow. And there is a good demand for this molecule. At least about 35,000 metric tons has been produced in the year of 2023, but natural menthol is in demand. So how do you reach this natural menthol demand? By increasing the agriculture productivity alone can increase the productivity of the menthol as well. And of course, due to this, the scope for the demand of synthesis of the menthol is being, practiced. The major companies like Simraisu, Takasago and BSF have been introduced to make this molecule synthetically by industrial production. And as you understand that, if you look at this one chemistry, so chemical synthesis is possible based on the simple chemistries are using the cheap raw materials to produce the molecule in higher concentration. There are a few the organic transformations which have been adapted by various industries, starting from the myrcene, metacresol, or cetral, etc. And various the smaller transformations have been adapted onto the molecular framework depending upon the final molecule, which needs to be developed in such a way that 1R, 2S and 5R, the menthol has to be achieved, and there are a lot of trade secret or the synthetic secrets in which the catalyst which is used for this production is being adapted or patented by various companies which are already mentioned. The beautiful nature of this the menthol could be seen in terms of the product applications, but when used the molecule in higher concentration, suppose if I want to have a higher the coolingness, higher the relaxation effect into the consumer product, it is very difficult to have it, because after reaching 30 ppm concentration of menthol, it will be having some kind of irritation to the consumer products. In order to solve this type of the irritation or a kind of increase the cooling nature of the molecule, people have started working on to making the derivatives or increasing the molecular library. In the literature, at least you will see that more than 2000 variants of menthol has been known. By utilizing single hydroxyl group which is present there, all the 2000 molecules have been synthesized to make menthol less irritation and more product applicability. In this case, so utilizing the hydroxyl group and making the amide or making the spiroethers or making the continuous carbonate esters has been tried, but very successfully the company which has been able to produce the molecule which is called coolant agent 10, which is a alkoxy ether between the glycerol and menthol. This molecule is called coolant agent 10. It takes over the various the productions in terms of the product applications. Today, if you see in the consumer products like the paste or the shaving gel or a lipstick or whatever which you use for a consumer products, more often you will see that the coolant agent 10 than the menthol do decrease the irritation property, and in this case this molecule has got the thousands of the millions of dollars for the company. So looking at this, the success story of the coolant agent 10, we wanted to make whether we will be able to see the various kinds of the derivatives of the very close isomeric menthols. In this case, menthol is having the saturated alcohol system, whereas the thymol and carbachol, first two examples which are given, they are phenolic compounds, but they contain only a double bond within the ring. Whether they will be, they are called isomeric to menthol, and we wanted to make a similar kind of the structure expansion in order to meet whether we will get a coolant agent kind of a molecule. In this, the construction we are able to do using a thymol and a caracrol as an example. So using a glycidol ether, the glycerol-menthols and then esters of the glycerol-menthols have been also constructed. These esters of glycerol-menthol are called as smart cooling agents. Smart cooling agent because we have the diacyl group. Diacyl groups can be cleaved by a lipase. So when the lipase is cleaving this one, you will get a release of this cooling agent. This is the one of the recent works from our laboratory. Similarly, so that we will be able to construct a molecular library of the thymol with the carbon atoms starting from the 8 to 10, 10 to 12, 14, 16, 18, etc. So that the molecular library of diacylated thymol, which are smart cooling agents, has been constructed. Similarly for the carbachol and also the various kinds of the C8, 10, 12 and 18, 16, all other the variants which are diacylated carbachol and diacylated thymols have been constructed, which are called the smart cooling agent depending upon the library which we are making. Further on the increasing the molecular library which has been seen in the glycine derivative of the menthol has found to be a TRPM8 agonist kind of a molecule, because TRPM8 are the enzymes which works in the range of 2 to 28 degrees centigrade, and the molecular variants which are also being used for this TRPM8 are found to be the amino acid conjugates. In this direction, all the amino acid conjugates have been successfully conjugated with the menthol with various kinds of this the molecular framework. When we are achieving some success in in building out the library of the molecules, we will be able to get at least one or the other lead which gives you the kind of the lead optimization and lead the generation to the commercial production. And moving on to the other sesquiterpene molecule which is being utilized in my laboratory, which we call as a sustainable laboratory, which is from the ginger which is called medicinal ginger, which is Gingiber zerum beta. So regular ginger which we consume in the food, which gives you the pungency, but if you use the similar kind of a ginger belongs to the same family, if you use this ginger in the food, it will not give you any pungency to the food, but it will give you the medicinal effects in terms of the stomach problems or a antipyretic or a anti-inflammatory or whatever the kind of this. Though it has been practiced in various places like in Northeast, in Kerala and other places, this ginger has been used for various the medicinal applications. This ginger is called the shampoo ginger or a medicinal ginger. So way back about 15 years back, we brought this ginger from the University of Spices Research Calicut, and from then onwards I started growing in front of my laboratory, and today we grow every year at least 200 to 300 kg, 10 mg of this molecule, which is a sesquiterpene molecule, which cost about 5 to 6,000 rupees if you procure from the Sigma Aldrich. So now we are a self-sufficient laboratory. We have produced more than dozen papers out of this, the molecule alone, and we are able to produce this molecule in about more than 200 grams. And 2 PhDs have also been completed, because otherwise I would have been investing more than 4 to 5 crores to complete the work which we intended to do, but we started growing this ginger and then extracting from the Gingiber zerum beta the sesquiterpene molecule zerum. so that the self-sustainability in terms of the laboratory could be achieved through this molecule. And the obvious reason of utilizing this molecule, because of it is having a, it is called NMRDOS, natural material diversity oriented synthetic material, because it is having a various several the medicinal effects, and also it is considered to be a nutraceutical molecule. Looking at the topomeric application of this molecule, which is having a like a V-shape, distorted V-shape molecule, so which is also connected with the dienone system in the molecule. So we wanted to whether we can increase a dienone to trienone, because the increasing the diversity in the molecule will also influence on the quality in terms of the bioactivity. So which is being thought about it, and we wanted to construct a molecular library based on the dienone chemistry which is present there. So we wanted to utilize the other part of the molecule which is less reactive, which is about 4 or 5 allylic centers. We wanted to do selenium dioxide oxidation on this molecule, so that we will be able to get a trienone system. Nature creates a dienone system, whether we can introduce one more system which is called a trienone, which is being able to possible with the introduction of the dienone to trienone, whereas the two molecules after this reaction could be able to obtain, which is one is alpha beta unsaturated aldehyde, other one is alpha beta unsaturated ketones, so that we were able to get increasing the molecular library. And towards the making one more the introduction into the molecule which is being taught or which is being introduced in the drug discovery chemistry, so which is the presence of CONH bond. CONH bond is ubiquitously found in the nature and also very much tolerable under physiological condition, and also today about 30% of the drugs in the world will be having this type of the CONH bond. So we wanted to just introduce the CONH bond into the cyclic ring, so for which we have used the Beckmann rearrangement reaction. Under this Beckmann rearrangement reaction, what we got is introduction of NH not only to in the ring, not only on the left hand side of the carbonyl group, but it also introduced the NH into the right hand side. Instead of getting CONH, we also got NHCO into the molecule. So once we have tried these molecules for the bioactivity side and the anti-diabetic the work, we have found that azazerumbone 1, which is being NHCO, is more active, and this is completely lost its activity. Based on this one, we were able to get a structure activity relationship on the molecular variants which have been made on this chemistry, and which is being shown here, and we are able to get the few molecules which are having the higher the strength than the natural material, or will be having a molecular diversity which is increasing, and which are being tested for various other the applications. Additionally, looking on the natural colors which is being in demand nowadays because of the rhodamine, the side effects on the children or the people who consume the junk food or the street food continuously with the where people use the synthetic colorants as the main ingredient to attract the food. But we wanted to whether we have any kind of the betalin can be introduced. When we talk about betalins are from the caryophyllase family, and the molecules which are responsible for the color are the beta xanthines and beta cyanins, which are yellow and red in color respectively. But this one, so suppose if you take a beetroot color as a natural food colorant, which is having a lesser stability to higher temperature and also to the variations in the pH conditions. When we thought about this one, we found a new source or a source which is called the globe amarantha. Globe amaranth is a flower which is being used in the temples, and in the season of maybe it will be produced during in the month of July to August or September. So this is having a beautiful pink color is the molecule, which is due to the beta cyanins, which are belong to the family of beta betalins. This is the success which we have achieved recently on extracting the color. So it is being worked on the pilot scale about 100 to 200 kg scale. We remove the fiber part as a good quality cellulose fiber, whereas the color which is added, which is extracted into water, and then the volatiles have been collected, and also plant growth promoter which triquintanol is also taken out from the petals as well. And this is the colorant which is used in the jellies, which is self-stable for at least 30 days, whereas this is the synthetic colorant used, the jellies which has been shown here, which though gave you the better the colors, but they are not very friendly for the consumption for a long time. And why only stop at this stage? Can we use the waste product or the dust which is coming out of the process? We also found that this is the example where the cotton candies have been demonstrated in our institute under the floriculture mission project, utilizing the globe amarantha for a coloring the food, and which is being shown there, and the left out material which is not able to be extracted can also be utilized for the making some of the holy color powders, and the students who have been applied and then played Holi with the waste which is being generated and then which is being used for the celebration of Holi. Moving on to the other natural colorant which is extracted from the Bixa orinella, which is a shrub, the molecule which is responsible for the bright red color is due to bixin, which is the C15 carbon atom molecule, which is having a ester group, and other end is a carboxylic acid group, wherein you see that in US it is allowed. And quite interestingly, why I am interested in this molecule or in this the productivity, because in Mysore climate condition this grows well. I have seen several places along the street the tree, but no one is interested to cultivate in the commercial production by encasing the agroclimatic condition which is present, which is the gift for the Mysore weather. So nobody has thought about it. But the process of making this colorant from the Bixa orinella is known in our institute for more than 3 decades, and the technology is also available. This could be one of the commodity which can give you the way of sustainable resource for getting the natural colorant, which is one need to look into it. And everyone is fond of this turmeric. Turmeric is valued for its the color, and the color is due to from the curcuminoids. The curcuminoids belongs to family of phenolic compounds. So one is the curcumin, D-methoxycurcumin, and bis-D-methoxycurcumin. And lot of the published data says that all the beneficial effects which have been documented here due to the molecule called curcuminoids, so which is being extracted from the turmeric, are realizable only in the laboratory, but intended to get the same level of the application in the body, it is not possible. This is due to because of the inherent nature of this molecule, which degrades within half an hour time. The half-life of the turmeric, the curcumin is very, very low, and it destabilizes after pH 7. In this case, can you construct a molecular library based on the diversity oriented synthesis and the target oriented? In this direction, we have been working on this molecule to make the diversity oriented synthesis utilizing the curcumin, and then we are able to get a water soluble, because curcuminoids are not water soluble. We can make them water soluble by connecting the covalently with the sugars, which is called a diglycosylated curcumin, which is highly soluble in water and has a higher the stability in the physiological conditions. Similarly, moving on to increasing the diversity oriented synthesis on the curcumin, we are able to make the various kinds of the amino acid conjugated with the Bach protected amino acid and a debugging process. Similarly, we have constructed the molecular library or the diversity oriented synthesis based on the curcumin chemistry to achieve various kinds of the amino acid conjugates, and also another molecule which is called a tetrahydrocurcumin, which is the white curcumin, in other words it is called, which is not having color due to the loss of the conjugation by hydrogenation process, and this also works beautifully in the biological system, and also various kinds of the amino acid conjugates, and the glucosides have been constructed on the curcumin molecule. Moving further on to connect the chitosan, low molecular weight chitosans are very much essential in the field of the in the medicinal chemistry, and we wanted whether the amine group which is present in the chitosan can be utilized to make the skip bases. So we thought very easy, but it is not going, and we have used a linker succinate and then constructed the molecular library of various molecules like curcumin, shogol, and then the gingerol. Moving back on to the ginger chemistry, which is the regular ginger which you use in food for the pungent purpose. And these are the molecules. Ginger contains the good molecular library of the phenolic compounds like 6 gingerol, 8 gingerol, 10 gingerol, shogol, 6, 8, 10, etc. and phenolic compounds. Okay. And these are all some of the processes which are available in CFTR that I am not going in detail. And this is the molecular structures of the gingerols which are being, the like a 6, 8 and 10, 6, 8 and 10 the shogols, gingerone, paradols, like this is a complex mixture of the molecular framework which is being present in the ginger oleoresin. So when you want to have a single molecule in a pure form, it is very difficult. Again, you have to go through a lot of the isolation and then the purification process, and which is rather than I thought can we construct a molecular library of the ginger. So based on the molecule which is eugenol. Eugenol is again present in the clove, but essentially in about 60 to 70% in the essential oil. We started working on this the allylic chemistry which is present there, and to make sequentially the molecule and arrive at the gingerol. In this case, nature makes a gingerol in 6, 8 and 10 2 carbon atom system, 2 carbon atom addition in a single step variations. But when we do, we will not only increase 2 carbon atom system, suppose you can add a single carbon at a time. The one is based on the like a 5 gingerol, 6 gingerol, 8 gingerol, or 7 and 9. These variations as in the synthetic laboratory you could be able to achieve, while biosynthetically plant achieve 2 carbon atom system in a single step. And of course, this is again the isoxazoline chemistry which is being worked out on making the gingerol more useful, and this is only the structure activity relationship wherein the bioactivity rests on the beta hydroxy ketone moiety while the activated aromatic system. So in order to make a variations in this the phenolic hydroxy, so we have to protect this one through isoxazoline or the moiety, and which is being tested, and also we are able to get this. Again, these molecules are not soluble in water, but they were made soluble utilizing the glucoside chemistry, and we are also able to construct the diversification in the molecular library which we wanted to dupe or cheat the bacteria. Bacteria communicates through a molecule called acyl homocidone lactone, which is of similar kind of these various the library which is being worked out there. So whether we can cheat the bacteria in such a way that it should stop in communicating. Suppose if you stop communicating, there is no biofilm formation. In order to break the biofilm, we used all the ginger bioactives which are having almost resembling the structural features. So we are able to get a molecule or library, so like a shogol, the aja shogol which is being introduced for the first time, and the gingerol and various gingerols which we have synthesized, tested against this anti-quorum sensing inhibitory activity to avoid the biofilm in the molecule. We can increase the molecular library of any molecule which you have in good quantity in the laboratory, or if it is not available, you make the molecule in your laboratory, so that whether you will develop the beautiful chemistry there to make the molecule, or use the diversification to test its the molecular library for various the beneficial effects. In this matter, we are also able to interested to make not only kind of a molecules which are just kind of the small insertion or the variation is a change, but also we wanted to make whether we can make a chemistry based on the enzymes, so that enzyme esters could be synthesized, which are not only important in the diffusibility in the various the atmospheric conditions, but they can also look like a kite. So artistically we can call as the molecule as a zerumbone oxide fatty acid kites. There may be a number of the variations into the molecules we can bring in, unless until you build a value into the molecule, it is very difficult to unless you have to make some kind of the changes which needs to be addressed, a challenge or a problem in terms of the solubility, stability or whatever this kind of. So in order to reach the sustainability, there are some good examples which we have seen, like the variations in the vanillin to make a ethyl vanillin to increase the threshold value of flavor activity, which is being achieved by the either with the agro sources or meeting challenges by the researchers, which are also able to get a waste utilizations, and then industrial manufacturers to meet the growing demands, and agribusiness operators to reach to the society. So there are some quite a big examples like ethyl vanillin, menthol to glyceromenthol, which is being talked about, and additionally still there are good number of problems available, like a curcumin stability under physiological conditions and economic isolation of 6-gingerol, or nutraceutical or chemical isolation or bar synthesis, which is the non-natural food colorant, which is one of the most triggering factor for the introducing the natural colorants for the food. At the end, so it is the integrated approach by the agriculture for the farming the community to help in getting the more sources for these molecules or the colorants or the flavorants, and economical processing practice to achieve by the scientists to provide R&D solutions, and scale of feasibility by the technocrats to increase the technology readiness level to higher the tone, and also commercial production by the industry, so which are very much essential, and circular economy to utilize the byproducts generated in the process, and agri-food business operators to reach to the society. At the end, it is an integrated approach by every individual which have been listed out there to get the societal, the sustainability. And thank you very much for your patience. And if you have any questions, I will be able to take.
Nice presentation, sir.
Thank you.
You took us to a world of chemical molecules with a small change in atoms, a group of atoms in the molecule, which alters the entire property of the molecule. Thank you so much for your presentation, sir.
Yeah,
Now
this
the
is
session is open for the questions. Anybody any questions?
this is my team who has been responsible for making all these molecules, and this is a very recent team, and big thank you all. Any questions?
Thank you very much.
Thank you.
Speaker with thank you. Maybe it's okay.
I thank Dr. Vidya B. sir for presenting his talk on understanding the scope beyond cultural
ಏನಂತಾರೆ
ಸುನೀತನ್ ಕಳ್ಸಿದ್ವಿ ಮೈಕ್ ಹಾಕಕ್ಕೆ ನೀವು ನೋಡ್ಕೊಂಡಿದ್ರಲ್ಲ ನೀನು
ನಾನಾಗೈತಲ್ಲ ಏನಿಲ್ಲ ಇದು ಅವ್ರಿಗೆ
ಹಾಕಕ್ಕೆ.
ಜೇಬಿಗೆ ಕೊಡು ಇದು ಅವ್ರ ಕಾಲರ್ಗೆ ಹಾಕು
ಇದೇನಿದು?
ಅದು ಎಕ್ಸ್ಟ್ರಾ ಅಂತ ಇದನ್ನು ಹಿಂಗೆ ಜೇಬಿಗೆ ಹಾಕು
ಬಾಯಲ್ಲಿ ಹ್ಞೂ ಅಲ್ಲಿ ಈಗ ರಕ್ಷಾ ಬರ್ತಾಳೆ ಈಗ ಹ್ಞೂ ನೀನು ಬಾಯಿಲಿ ಇದು ಮೈಕ್ ಹಾಕುವ ನೆಕ್ಸ್ಟ್ ಅವ್ರಿಗೆ ಹ್ಞೂ ರಕ್ಷಾ ಹೋಗಲ್ಲಿ
ಅದೇನೋ 5 minutes ಡೋರ್ ಓಪನ್ ಮಾಡಬೇಕು ಅಂತ
ಯಾರಿಗೆ ಅವಳು ಇಟ್ಕೊಳ್ಳಿ ನೀವು ಹಾಕಕ್ಕೆ ನೆಕ್ಸ್ಟ್ ಅವ್ರಿಗೆ ಅವಳಲ್ಲಿ
External heartfelt thanks to Dr. V.K. Swami, Sir, Associate Professor, Department of Chemistry, Jesus College of Arts, Commerce and Science, Nandigood, for chairing the session. Thank you, sir.
ಇವರು ನೆಕ್ಸ್ಟ್ ಯಾರು ಅಕ್ಕ ಅಕ್ಕ ಅವ್ರು ಯಾರು ರಂಗನಾಥ್
ವೈಟ್ ಶಾರ್ಟ್ ಅಲ್ಲಿ ಮನಸ್ಸಲ್ಲಿ
ಸರಿ ಅಕ್ಕ ಇವ್ರು ಹೋಗಿ ಇಟ್ಕೊಂಡ್ಬಂದಿಲ್ಲ ಈಗ
ಇಡು ನಿಂಪಲ್ಲಿ
ಇಡು ಅಲ್ಲಿ ಚೇಂಜ್ ಮಾಡ್ಬಿಟ್ಟು ಬಾ ತೆತ್ಕೊಂಬ ಇದು ಇಟ್ಬಿಟ್ಟು ಇವನೇ ಯಾರ ಹತ್ರ ಇಟ್ಬಿಟ್ಟು
Now we'll have the sixth invited talk from Dr. R.R. Arun Ranganathan, sir, Assistant Professor, Amity Institute of Biotechnology, Bangalore. I request Dr. R.R. Arun, sir, to take the seat on the dais.
ವಾಟರ್ ಬಾಟ್ಲ್ ಎತ್ಬಿಡ್ಲಾ ಅದು ಎತ್ಬಿಟ್ಟಿರೋದು ಸೊ ಪ್ರೆಸೆಂಟೇಷನ್
It is a great privilege and honor to formally introduce the chairperson for this session, Dr. Suhas R. Sir. I welcome you, sir. Dr. Suhas Sir is a senior scientist in the Department of Plants and Products, Spices and Flavor Technology at CSIR, CFARI, under the Ministry of Science and Technology, Government of India. He earned his bachelor's, master's and doctorate degrees from the University of Mysore. His scholarly contributions span from chemistry, spice bioactive, peptide and medicinal chemistry, and conjugation chemistry. He was the recipient of the prestigious NRF DST postdoctoral fellowship at the University of KwaZulu-Natal, South Africa. With over 16 years of teaching and research experience, complemented by industry exposure, Dr. Suhas Sir has published nearly 65 scientific works in reputed journals with a notable H index of 22. He has organized and participated in numerous state, national and international seminars, conferences and workshops in various capacities such as coordinator, convener, organizing secretary and committee member, earning several awards and appreciation. His research papers have been presented across the USA, Germany, Australia, South Africa and India. We are deeply honored to have an eminent scientist of this stature chair this session. I respectfully invite Dr. Suhas Sir to kindly proceed over the proceedings.
take some time because
A very good afternoon to all. The last of the academic session, I have a great pleasure in introducing the speaker, Dr. R.R. Arun Ranganathan, who is an assistant professor in the Department of Biotechnology at the Amity Institute of Biotechnology, Amity University, Bangalore. He holds PhD in microbiology from University of Mysore and has extensive academic, research and administrative experience in the field of life sciences and industrial biotechnology. He has been associated with major funding projects by ICMR, DRDO, etc. He also served as visiting researcher at Cardiff University, UK. His research expertise spans industrial biotechnology, green nanotechnology, phytochemicals, food and environmental biotechnology, and many others. In addition to his research, he is also involved in teaching, mentoring PG students, laboratory management, as well as curriculum development. He currently serves in multiple academic leadership roles, including program lead for MSc BT and faculty coordinator for Genovate Biotechnology Club. His academic vision focuses on sustainable technologies, future food systems, industry-oriented biotechnology education, and inspiring the next generation of biotechnologists through innovation-driven learning. We are very happy to have you, sir, on this occasion, and now I request you to take over the session.
Thank you, sir. Am I audible? Am I audible?
Written.
Yep. Am I audible, sir? It's not on. Okay. Thank you. Yep. Okay. Thank you. Am I audible? Okay. So, very good evening. Anyway, we are most ahead the time, and I'm not exactly the subject expert in food. I started working on this. So we got a scenario over there in my master's laboratory students experiments. We got a scenario to create a nanoparticle which can be edible like. So we started the work related with the food science. So my presentation today, I'm going to make it which can be interactable like, because it is not the research work, any related research work which I'm going to present like. So I wanted to make little bit interaction which I can make it, and what can be the future which the chemistry people can involve and develop, which your contribution, where exactly requirement is there for the food. Okay, so you all aware food is coming only from agricultural background, like only is it possible to synthesize the food in the laboratories? Because my title, I have started my title with food from laboratory to plate is the title which I have given. So do you have any idea we can produce the food over there in the laboratory? Anyone have an idea? Anything have you heard such like? Okay, maybe I need to make a little session break. The speak which we are making, if the people are not there over there in the attention, to make the attention, there is a trick is there. There are 3-point trick like. One is start with the politics or gossip. So it is easy to interact. The interaction will start. The unknown person, if I'm first time I'm meeting, I could able to initiate the interaction. So I don't know the person completely. If I want to make the initiation, first thing. So we are students, we don't want that. The second one is food. Ultimately our title is related with food. And third one is movie. So if I start something related, talking about movie, I may get the interaction, maybe I may get the answer. If I ask a question, I may not be getting. So how many of you seen Jurassic World movie? Okay. Jurassic Park, maybe 90s kids only know that. Jurassic World is for your generation. Jurassic Park, how the dinosaurs are created in that movie, any idea? They start from where exactly in that movie? They tell one story. They used to get the mosquito blood over there. They used to get the dinosaurs DNA and they start like. So such like in 1966, one movie is there, Star Trek. Over there in that movie, there is a mission which print the food, whatever we wanted like. So if I want a burger, the mission, if I make the order, it will print the food for me. The concept is 1966. The movie's concept is 1966. But now we could able to achieve. In 1992, the Jurassic Park first movie came. Are we achieved that state? Can we get the animal from DNA? Do you have any idea? Okay, anyone heard colossal biosciences? Anyone heard colossal biosciences? Dire wolf. Anyone heard in recent days over there in last year? Okay, which the company Colossal Biosciences who achieved 3000 years old extincted animal who revived over there now in current state. So in 1992, which was the movie concept, now it is emerging. The Colossal Biosciences, which they have the ultimate goal is they wanted to make mammoth, which is the one ice age mammoth which they wanted to revive. That is the goal which they wanted to. If you are cross-checking over there in the internet, if you check the colossal bioscience websites, they are having the huge demand over there in current scenario. They are getting the multiple agencies who are funding anything like the money to de-extinct the animals, because so many animals are getting de-extinct because of so many environmental parameter and such like. They have so many parameters. In that, one animal is called doo-doo bat. Have you heard anywhere? Maybe doodles or somewhere else you might be heard or seen. Doo-doo bat is the one which in Iceland, Madagascar, Iceland, which got extincted only by human. So when we entered to that Iceland, we completely eaten that bird, means not us, our ancestors, who has made that animal completely extincted. So now they are trying to revive, because that animal meat is that much tastier one. So such like, if we are printing whatever we want to make the animal meat to prepare, we can prepare like. So it is not there over there in a conceptual state. Actually it is there commercialized already. So if you check Milk or Imagine Dairy, these two are the company who producing the milk. These two are Israel based company who is producing the artificial milk completely by yeast, pichia and bacterial cells. By using that, they are producing the milk. The FDA clearances happened and they are supplying the milk over there in Singapore. So same like, there are two companies are there. If you check over there in internet, you could able to No Cow Factory or Zero Cow Factory, which is Punjab based company in India, who started the work to create the milk by bacteria. And Bangalore based company, one, they have started 5XX40. So that company also they started to make a milk by using yeast cultures. So maybe in soon after your generation, if you are in masters, if you are completing your PhD, you may be getting an option to enter into that company to get a job to produce the artificial milk. Okay, so do we accept such kind of milk? In case if it is produced by bacteria, will you accept it? I am telling Singapore it is getting accepted. So over in India we accept it. Why not? We don't accept. Okay, curd is a vegetarian or non-vegetarian, you tell me? Huh? Curd is a vegetarian or non-vegetarian? Still doubt is there, huh? Curd is vegetarian or non-vegetarian? Vegetarian only. So here we are producing the milk by bacteria. So it's considered like vegetarian only, right? And we are not killing an animal, we are producing the milk. Why can't we? We cannot take. We can. Okay, I'll give one example. Over there in 1980s, till that if we want the insulin, we are completely depending on pancreas of animals. So animals' pancreas only we used to get the insulin. Over there the dilemma changes also there. There are even the discrepancies also. It is from animal like. Vegan people, they were not able to. But now, which insulin we are currently consuming, the complete insulin is from bacteria. Anyone aware of that? Humulin, the insulin is actually produced by bacteria right now, which we are having in the market. Okay, so the generation as which I have discussed, like the Singapore, which the country which has adapted the technology, Gen Z means your generation is the one, they wanted to taste it and they have accepted it as per the statistical which they are telling. So maybe in India, maybe your generation is the one who is telling we are not ready to take. Properly when the company is coming, you may be a part of it to produce, or otherwise you may be a part of a person as a consumer to consume it, as per my knowledge. Okay, so there are multiple ways to make a food over there in a laboratory. We can print the food, we can ferment the food, and whatever the nutritional composition we wanted, like such a way the work is happening, like since which I discussed about the Milk, the company. Casein is the main protein which is present over there in the milk. So milk components they are creating by bacteria by the fermentation. A simplest formula to understand a non-biologist, it is nothing but the components what and all present in the milk. It is produced by bacteria by fermentation. Okay. Yeah, so the demand is happening. So in 2050, 2034, so many predictions are happening. Like right now, what the consumption of meat we are consuming it will not be a sufficient one to make the production in a very shorter period of time. We want the high. Sorry. So 6 months, which used to take over there the native breed to grow, now we are more than within 40 days we could be able to achieve. The meat growth is happening like, and it is not achieved by genetically modified organisms. It is by crossbreeding. Right now, which we are there in the state and which we adapted up to the level of crossbreeding. Maybe the next possibility is there to adapt to artificially created like. So probiotic drinks, any examples can you give? Probiotics. Curd is a probiotic. Yogurt. Over there in the markets, you could be able to see the sentences which they are marketing itself. They are writing it as a probiotic curd, probiotic milk like. So nothing but over there, when it is coming to yogurt state, it is not the traditional food of India. So over there, the cultural based variations, the different kind of microorganisms are present over there in the food sample for the fermentation. Based on the microorganism count and what the source they are utilizing, we are naming them. So curd here which we are utilizing is a normal milk, and 1 or 2 strains of bacteria will be there present over there for the fermentation. So when the bacterial count is increasing, so that time we used to get the different namings. So right now, the people's stomach commensals loads are getting less, and stomach related ulcer and other problems which it is increasing. The food supplement which they are providing is probiotics only, which is nothing but the multiple organism mixed food which is commercially prepared, which is marketed over there for the treatment of stomach related problems. So stomach and your anger related research is also happening like. So multiple organisms which is mixed and already commercialized over there in the market by the name of probiotic. So little modifications. You all familiar with Yakult? Anyone? Have you seen over there in the supermarket? So it is a probiotic. They used to tell 180 million cells per shots. So entire bottle is actually a patented design. So there which strain they are using, it is actually a strain which is a slightly modified strain like. It is isolated and patented strain. So we are accepting it and we are not knowing aware like we are actually adapting the modified organisms like. So maybe in the future, these things also without knowing we will be, because the company is there over there in Gujarat. One or the another time, the product which we are utilizing, we may not know the milk came from where. So properly if you check the ingredients over there in the Nandini milk packet or whatever, the milk solid. So if you properly could be able to see the ingredients, you could able to distinguish. So the milk production which already we are commercial product which we are using itself, it is having so many artificialness. Okay. So here the comparison which we needed like. So instead of normal agriculture, we will be making it like. So as a chemistry point, it's nothing but food is equivalent to molecules. It's a proteins, lipids, carbohydrates, metabolites. Those things if we separately create them, then it is fine. It is possible to make it as a sustainable supplement to compare as a food. So this is actually a reference paper only. Since I wanted to explain to Gen Z, I utilized completely AI only. So the data is mine, but the presentation is not by me. Yeah, so plant-based supplement is also there, plant-based meat is also available. Now, vegetarian, non-vegetarian to compare, veganism is also getting grown. Over there they wanted to get the meat which is equivalent to, which the meat is prepared by a vegetarian and it is equivalent to the taste of non-vegetarian, how we used to get it like. So such based foods are also already available. This is also similar way how in the industrial fermentation is happening, such way produced meat only. Okay, so here these are all the example companies. If you enter into internet, you will get to know their details. So they are already approved companies which they are there already. They have marketed over there in Europe, America and Singapore. Even China also they have adapted the artificial meats. So nature's friend, here friend spelling is there in a different way, FIND, and Evrico. This is the company names which you could directly search over there in the internet, which they are producing the artificial meat. So more than 175 companies are there globally. In India, we just started with milk, not meat. Milk companies, there are 2 are there, already there in the startup state. So how we used to make the meat get ready? The simplest process is we have to get the biopsy sample from the animal and we have to make the bioreactor into multiply into the high scale production. Then we have to differentiate the cell which we wanted to be. It will be made by the scaffold support to meat, how to create the meat, like what exactly the meat we wanted, like not the bone. With the scaffolds basis, that is also made up of food graded materials. The final meat is ready. It's not like which the diagrammatic, like it's already present over there in the market, and the sensory taste is also similar, like the artificial meats taste is also similar like how the normal meat is tasting. Yeah, so the scaffold layer is also made up of plant or animal based materials which is created over there in the laboratory conditions. Okay, so this is the company Good Meat 3, so which they commercially already they made it and they are already there in the markets in 2024 onwards. Now we are in 2026, the meat is already there in the market. So they are even cost wise also day by day, the Moore's law which they used to apply over there in the IT for the processor to grow, the same way the meat price is also getting less. When the invention happened, that time the meat price is higher than the normal meat price, but now it is getting into the reducing state. So even 2013, which was done by a laboratory research only, the first burger which created over there in the complete laboratory. So over here, it is a different company who made as a meat, but all point it started as from 2013, a company who created the burger, meat burger over there in the laboratory. Yeah, so CAGR, compound annual CAGR, growth rate, they used to tell over there in even in economy rate to tell how much the demand is there and the growth is there. So this is having very bad demand in future and the growth is also there. And even Indian based startup companies also there, so we can be like, so here the precision fermentation is the one which is the principle exactly behind the milk, which before I opened the slide which I made little interaction, that is the formulation which we use to create the milk. Artificial milk is a precision fermentation. How much casein should be? Some people they have a lactose intolerance, that not there before in India, but nowadays it is there in the increasing state. Like even India is also the cancer capital. In a recent data, the last 25 data was India is having the highest cancer patients rate last 5-10 years, but last year data is drastically which is there in a high state, more over there in Tamil Nadu, Chennai I think. Over there the report is released like in such a state, we are having the more cancerous, like even because mostly we can say as a food habit. One of the thing, the food color which we are using is also a very dangerous thing like. So practically the principle for the precision microorganism precision fermentation is over here we used to separate the proteins which we needed. It will be converted as a dairy products or direct supplementary which is required for the dairy based products which is made as a bakery cake and all, like over there the ingredient instead of milk to use, the synthetically produced milk can be addable over there. So as I early mentioned, like in 2034 we have a high demand of artificial milk which will be having a highest value like. So this is also I already which I told like, so we have accepted the insulin. The similar cases also happens over there for our artificial food also. We may tell like in the beginning we may not, but we are adapting most of the food items such like only pizza, burger. No, Indians we don't eat like, but we are the highest consumer now. Same cases, chicken or whatever the kebab which colored, no, artificial color I never touch, but that is the thing, the color which is banned over there in India also, but every shops you will be able to get that color. So such conditions will create, we are the highly populated country, and when the demand is there, the need is there, the production is also will happen. Yeah, so these are all also the company which is already commercially available. Brewed cow, ice cream company is there, actually that E is not in a proper way. Maybe AI made a mistake. I didn't. Yeah, modern kitchen, cream cheese. This is also there as commercially available product, which is created artificial by using artificial milk. So the sensory validation, which happened widely, and FDA approved thing also happened over there in the sensory. The person which has given as an artificial and non-artificial meat, while tasting they couldn't able to distinguish which is synthetically created, because both of them are similarly tasting. So over there your chemistry will play. In 2015 they have created a AI which is called NATCO. That time it was not an AI, it was a machine learning program. Which formula if we are providing for the food, exact taste will come. So some kind of rusty smell or the taste of the entire meat or the milk you want to provide, what exactly the compound you have to create and you have to add it to that particular product to get that taste. So this is the company which they have created over there in 2015. They completely analyze and what ingredient to be added to create the smell. It is for your sensory. Okay, this particular AI they have now it developed up to the AI state. They are having a huge database regarding the tastes. Okay, yeah, so here is an example how we can create the heme taste. So the formulation to make by using the AI help, how we can create the taste as well as the color for the meat. Okay, yep, so 3D printing way is also there. That is now in the level of cake, not exactly to the meat. Meat synthesis is happening, production is happening, but not by the way of 3D printing, because it is becoming more costlier to compare synthetically creating milk. And artificial meat, which till now I have discussed it, we are utilizing a cell which is retrieved from the animal and we are creating it. But from here I am utilizing the fungus, which is nothing but Fusarium or some specific species are there. They are helping to create the meat texture which is made by fungus. Here the meat is completely based on the vegan product, like even the fungus, mushrooms, some people they consider as a non-vegetarian. More debate is there for that. Mushroom considerably as a vegetarian or non-vegetarian, so I don't want to go deeper in that, but this comes under fungi. So here we are creating the fungus to create the artificial meat. Okay, yeah, so artificial fungal based meat products which is there commercially available over there in the market is meaty and nature friend, before which I have discussed. These two are the product which already available commercially over there in the market, which is produced by fungus as created as a meat. Yeah, so protein source, this is known factor only, where exactly we are there in the demand. Spirulina, so long we are utilizing for the protein dependency, even the space food or superfood which they used to tell is a spirulina, that also can be modifiable and integrate the bacteria, fungi and algae to create a specific food which is required for the particular person, a person who is having a hair loss, a person who is having a skin tone change. So what are all the nutritional supplements nowadays we are artificially taking as a separate sources, it can be integrated into food. The newly emerging area is there, nutraceuticals. So over there that also can be dependable and integratable when it is coming to industrial way of producing the food. If we are producing the food instead of normal way of agriculture, if we are customizing them completely, then we can completely customize based on the nutritional sources for the individual persons also. Yeah, so kefir, which is popularly known fermented metabolic health product, which is having the bacteria and yeast association. More than 16 strains of microorganisms are present over there in that specific curd. So such like the microbial associations are there in between bacteria and fungi to produce when the required amount of nutritional values when we wanted in the increased manner. So the customization is already there. We are just adapting them to make it for the sustainability, where now and in future we are getting the demand. Okay, so this is what which I discussed, like the person is not having a proper digestive system, like, so we have to customize the probiotics for the particular person. So in the way of producing the food, we are actually providing the medicine for the person. So in ancient days they used to tell good food as food is the medicine, like you will not get any disease. But now we are providing the medicine as a food. We are there in that state because we are already there in so many problematic health conditions only in last few years. Most of the things are there related with health conditions. So it became reversed in 30 years before, which we consider good food is a good health. Now good health to make medicine we are converting as a food. We are reversing back. Yep, so personalized thing like, and it will be getting monitored like this, not exactly related with food. Right now, whatever the AI and other technologies are there, if we are integrating all of them into one roof, so we maybe get to, all the things can be controlled by only by food and nutrition which is required. The entire health is depending on your food. That food can be controlled in your laboratory. When it comes to your plate, then everything is solved. So demand is there, as well as same time your part of contribution is there more. So if you know to make it, so 1 or 2 startup companies are there. When you complete, maybe you may be a person who can do the startup company also, if you understand the principle completely how we can make it. So as the statistics data which I earlier discussed, like, so Gen Z is the one who adapted this technology more to compare boomer generation. And some they have adapted, 58% of people, they have adapted this technology because they are not familiar with what the terminology we are telling, artificial meat, they don't know what it is. Without knowing that, unfamiliarizing with the terminologies, the people adaptation is also happened in the statistical way. Nearly 58% of adaptation happened. Yeah, so as I said, like in future, the more adaptation will be there by your generation only, whom right now in the audience who told not much, you will be the persons who will be there in the adaptation. Once the regulation and trust is there, these two processes are going on. If these two are there, then you will be getting adapted to the technology. Yeah, so this also I have already discussed how we have adapted, but it is not right now the topic discussion which I am making. It is not completely artificially made. It is actually made by the selective breeding. It is actually by the way of breeding we achieve that which we are having in the market, which grows 4 times faster than the native breed. Yeah, so as I said, like biologists are having over there in the development state, they need more engineering to create a specific person composition. Chemists are need for the entire process to make it, and which I have discussed related with the flavor and other things to generate and make it over there to make a combinational food. And data scientists requirement is also there. Completely AI dependent world like only. So maybe my guide generation for the reference, they will be going library to library. Pondicherry library, I have got one reference like. My generation, we may be getting Google Scholar. You people are ChatGPT generation. So you are instant. So you may be more utilizing the data structures instead of references which we have to go for the library. So data sciences also required. Already you all are familiarized with your hand, which every day you are utilizing like. Yep, so maybe your future in 2050, if I alive or not, I don't know. But if you people are alive, your breakfast will be ready. The milk will be ready by already which is commercialized. The meat, if we adapted over there in India, that meat is also will be there in the plate, which will be created by artificial way only, which may be produced by you and consumed by you only. So over here, I'm just concluding. We have the entire session is mostly related with the research developments which is happened over there in the particular laboratories. I'm just now entering into the field of food science research because I'm not a food science person exactly like. I'm just now entering into the field. I have an interest what exactly happening over there into the world. So I just searched. You may be known by Instagram post or something like. I just thought maybe I think I made it completely new. Some GK question informations like I made, I think so. Maybe it will be useful for your future to decide which the food is also having an important chemistry role, which you can utilize it and you can learn. Immediate after also you have a job opportunities related over there in the food, not only by the way of testing, because those are all the things classical way everyone knows. we wanted over there in the future make it to sustainable. These things are all the required things which you should know. At least you should aware of. These type of things are there. Okay, so over here I'll just conclude my talk. I don't want to make it more. Yeah, so if you have any questions, you can ask me.
Hello. The floor is open for the questions. We can have a couple of questions.
So you need to search Google for the questions, sir.
Alright, if you do not have any questions, I take this opportunity to thank you very much sir, for coming all the way from Bangalore and giving a wonderful speech.
Yes.
Thank you very much on behalf of JSS Science and Technology University. Thank you very much, sir.
Thank you, sir. Yes.
I thank Dr. R. Raghunathan sir for giving such an insightful talk. Thank you, sir.
Thank you. Thank you. Thank you. Thank you.
We'll take it.
Yeah.
I thank Dr. Suhas R. sir for chairing this session. Thank you, sir.
I think it's not off. Thank you. Slowly. I think I'm not able to get the trigger. Thank you.
ಸರಿ ಯಾವ್ದು ಕಾಲರ್ ಬೈಕ್ ಓಕೆ
Good evening, everyone. I request Dr. Bettadaya sir, Dr. K. Kendaganda Swami sir, and Mallu sir to take the seat on the dais. Feedback help ಬೇಡ. With humble pranams at the sacred feet of His Holiness, I extend a warm welcome to one and all to the valedictory ceremony of the One Day International Conference. Today's conference highlighted the vital link between chemistry and food sciences with the invited talks and poster presentations offering values, insights and meaningful academic exchange. With this, I now request Dr. Bettadaya BK sir, Senior Principal Scientist, Plantation Products, Spices and Flavor Technology Department, CSIR CFTRI Mysuru, to share few words with the gathering.
Namaskar, present here. And it's a morning, from morning to here, very wonderful session we had. So, 5 technical sessions and also the collaboration between the Hokkaido University and then the JSS Technology University, the signing of MOU and then taking the collaboration to the next level has been, we are seeing the technology function out of the 2 talks. From the Hokkaido University, Professor Siddhavasegowda at the beginning and then Dr. Stone the second session. And I very much appreciate the Professor Mallu sir, the organizing chair, who is a silent crusader with a happy face and then the lead from the front and the making success, taking the whole team into conference and then achieve success. And the success goes to him and then my appreciations to sir for conducting this wonderful the One Day International Conference here. And of course we have seen the couple of things which are very very essential for the younger generation to take a note on it. Either from the Hokkaido University faculty who have thought on the lipidomics research, which is very essential in identifying the various kinds of the lipid parameters for identification of any kind of health issues or kind of whatever other the relevant information which has been briefly and so meticulously explained by Professor Siddhavasegowda in the morning, and then followed by Thorough Swan, who has shown that how the grape the chemistry works based on this the various the inputs which are necessary to the microbiological applications. Followed by a session by our colleague Professor Prakash Alani, so who has also given insight onto the chemical the metabolites which is being produced by microorganisms, how they are important, and also explained on the various aspects of the the microbiological metabolites, how they will be able to produce various kinds of the molecule of very interest, and then very they also touched upon the probiotics etc. Followed by a session from the Santosh Gowda party, who also taken us to a session where how the coffee which is being brought out by Baba Udan into Chikmagalur way back about 400 years, and then now we are the leading of the producers and the consumers of coffee, and particularly both the varieties Arabica and then Robusta. So, and also the various stresses which is being exposed by the coffee plantation for the adaptation or the kind of the environment which you have been gifted by nature to have the shaded coffee plants, which is one of the important aspects. So that is called a brown gold, which is being being taught by the party, followed by the lunch. Then I think my talk, which is being also focused on the bridging chemistry and then the food, understanding the spice molecular interpretation or molecular the prominence in order to take either the flavor or the color to the next level. So the younger generation here so need to apply an integrated approach to in order to live in a society where the sustainability is one of the prominent factor. And of course, I also thank sir to coin the word sustainability in this modern era, where the competition is one of the biggest the thing which is coming in all the Gen Z the way. One we have to prepare for how to the face it. Otherwise, the future of our the students and Gen Z is going to be very difficult, for which we need to have preparation from all-rounded approach, for which the conference has provided the inputs or the insights so that we will take it to a next level, so that self-sufficient Bharat or the Viksit Bharat, the dream of our Prime Minister could be achieved through various the ways in which this type of conferences where you talk on, learn things and adapt it into a very productive the conclusion in terms of the commercial production or the kind of a startup to grown into a, for example, if you take a ID batter, many of you may not be knowing how the ID batter is being grown. So ID is nothing but idli dosa batter, which is a technology developed in CFTRI, which has been transferred to one of the new entrepreneur who is taken it to the next level and is one of the big brand nowadays. And like that, the commercial production or take away the research which is being there, which is being conducted into the market as for the commercial application is one of the need of the hour of the Viksit Bharat dream. And at the end, Professor Ranganathan has concluded in such a way that the lab to the plate. Lab to plate is, of course, today we are seeing may not be realizable very immediately, but this biotechnological approach has a really tremendous growth in the society as we adopt a Yakult into our product as a consumer product. We also hope to get at least a few products in the future line, so which will be able to supply a nutrition kind of the health product or kind of the requirement of the younger generation. In this direction, the kind of the arrangement which is being made by the JSSTU Technical University is appreciable, very commendable, and which has been guided by the whole team of the organizing this conference. I'm so thankful and really blessed to be here this morning. So the talks are being arranged very meticulously and also being rendered by the speakers. My only single worry is no student is coming out to ask questions. At least still the floor is open, I believe, if anyone will have any questions in this regard. It must be a very, very interactive. The enthusiasm which you have is driven, the driving us to take the session into the next level. That is many of the speakers have been felt. Of course, please learn asking questions. That is the thing which is need to be need of the hour as well. So I am very thankful to the JSSTU and also the organizing team for calling me for a talk on this connecting spice chemistry and the sustainability. And I thank you very much and special thanks to Professor Himas.
Thank you, sir. I now request Dr. B.K. Kendaganda Swami, sir, Associate Professor, Department of Chemistry, Jesus College of Arts, Commerce and Science, Nanjangoodu, to share a few words with the gathering.
Dignitaries, Andreas, Papadayas, and my dear students. This college, this institute is organizing so many workshops, seminars, conferences just for the benefit of the students. As Vedya sir told that you have to interact more with the resource persons, because then only you can learn the things. The beauty of chemistry does not lies in mere explaining the things. The beauty of chemistry lies in why, how, etc. Okay, therefore you must ask the questions, then only we can learn few things. Okay, with this background. Okay, now this department organizes so many workshops, so many seminars of national levels and international level. Okay, they are coming, the people coming from so many institutes, you know, and so many countries. From in the morning, the scientists from Hokkaido University, Japan, they came in the previous seminar. So many scientists from different countries, they used to come and visit this institute, and they are giving so many informations for the students. Okay, so you have to share your thoughts, and you have to interact with the delegates. Okay, with this I thank the organizers for organizing a wonderful program. Okay, thank you very much.
Thank you, sir. I now request Dr. P. Mallu, sir, Vice Principal, Program Convener, to share a few words with the gathering.
Good evening to one and all. Say good evening. ಸಾಕಾಗಿದೆಯಾ? ಆಯ್ತು. ಈಗ ಯಾರೋ ಕ್ವಶನ್ಸ್ ಕೇಳಲಿಲ್ಲ, ಅದಕ್ಕೆ ನಾನು ನಮ್ಮ ಮದರ್ ಟಂಗ್ ಜೊತೆ ಮಾತಾಡ್ತೀನಿ. You are please all of you interact with me. Because all of you are from Mysore, Chamarajanagar, ಈ ಸರೌಂಡಿಂಗ್ಸ್ ಅವರೇ ತಾನೇ ನೀವೆಲ್ಲ? Your mother tongue is Kannada. ಈಗ ಬೆಳಿಗ್ಗೆ ಸ್ಟೇಜ್ ಮೇಲೆ ಇದ್ದವರು 90% ಕನ್ನಡದವರೇ. ದೇ ಆರ್ ಆಲ್ಸೋ ಕಮಿಂಗ್ ಫ್ರಮ್ ದ ಸೇಮ್ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್. ನೀವು ಯಾವ ರೀತಿ ಬಂದಿದ್ದೀರಾ ಅದೇ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್, ಇನ್ಕ್ಲೂಡಿಂಗ್ ಮೈಸೆಲ್ಫ್, ಇನ್ಕ್ಲೂಡಿಂಗ್ ಬೆಟ್ಟದಯ್ಯ, ಕೆಂಗನ್ ಸ್ವಾಮಿ ಸರ್ ಇರ್ಬೋದು, ಇಲ್ಲಿ ಡಾಕ್ಟರ್ ಸುಹಾಸ್ ಇರ್ಬೋದು, ಬೆಳಿಗ್ಗೆ ಸಿದ್ದಬಸವೇಗೌಡ ಇರ್ಬೋದು, ಅವರೆಲ್ಲ ನಿಮ್ ತರನೇ ಇದ್ ಬಂದಿರೋರು, ಬೇರೆ ಯಾವ ತರನು ಬಂದಿಲ್ಲ. ಬಟ್ ಒಂದು ಏನಂದ್ರೆ ನೀವು ಇಂಟ್ರಾಕ್ಟ್ ಮಾಡ್ಬೇಕು ಅಂತ ಅವರೆಲ್ಲರೂ ಹೇಳ್ತಿದ್ದಾರೆ. ನೀವು ಮಾತಾಡಿ, ನಾನು ಬೆಳಿಗ್ಗೆನೂ ಹೇಳಿದೆ ಎಲ್ಲರ ಜೊತೆ, ವಿತೌಟ್ ಎನಿ ಇನ್ಫೀರಿಯಾರಿಟಿ ಕಾಂಪ್ಲೆಕ್ಸ್, ವಾಟ್ ಎವರ್ ದ ಲ್ಯಾಂಗ್ವೇಜ್ ಯು ನೋ, ನಿನ್ಗೆ ಯಾವ ಭಾಷೆ ಬರುತ್ತೆ, ಯಾವ ಭಾಷೆಯಲ್ಲಿ ಅನ್ನಿಸ್ತದೆ ಆ ಭಾಷೆಯಲ್ಲಿ ಮಾತಾಡು ಅಂತ ಹೇಳಿದ್ವಿ. ಅದೇ ನೆಕ್ಸ್ಟ್ ನಿಮ್ಮನ್ನ ಬೇರೆ ಲೆವೆಲ್ ತಗೊಂಡೋಗೋದು. ಈಗ ನಮಗೆ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಹೇಳ್ತೀನಿ. ವಿ ಆರ್ ಸ್ಟಾರ್ಟೆಡ್ ಇನ್ ಅವರ್ JSS STU MSc ಫ್ರಮ್ ದ ಇಯರ್ 2017-18. 2017-18 ನಾವು MSc ಸ್ಟಾರ್ಟ್ ಮಾಡ್ದೋ. ಅಲ್ಲಿ ಬಂದ ಹುಡುಗರು ಬೇರೆ ಎಲ್ಲಿಯವರೆಲ್ಲ, ಮೆಜಾರಿಟಿ 90% ಫ್ರಮ್ ದ JSS ಮೇನ್ ಕಾಲೇಜ್, ಚಾಮರಾಜನಗರ, ನಂಜನಗೂಡು, ಮತ್ತು ವುಮೆನ್ಸ್ ಕಾಲೇಜು, ಯುವರಾಜಸ್, ಮಹಾರಾಣೀಸ್ ಕಾಲೇಜ್ ಅವರೇನೆ. ಅವರು ಅದೇ ಹುಡುಗರು ನಮ್ಮಲ್ಲಿ MSc ಮಾಡಿದ್ರು. ನಾನು ಬೆಳಿಗ್ಗೆನೂ ಹೇಳಿದೆ, ಈಗ ಇಲ್ಲಿಗೆ ಒಂದು 6-7 ಬ್ಯಾಚ್ ಆಗಿದೆ. ಮೋರ್ ದೆನ್ 35 to 40 ಸ್ಟೂಡೆಂಟ್ಸ್ ಫ್ರಮ್ ಅವರ್ MSc ಸ್ಟೂಡೆಂಟ್ಸ್, they are pursuing their PhD in foreign university with fellowship. ಎಲ್ಲರೂ ಹೊರದೇಶದಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಕಂಟ್ರಿಗಳಲ್ಲಿ PhD ಮಾಡ್ತಿದ್ದಾರೆ. ನಿಮ್ಮಂಗೆ ಇದ್ದವರು, ನಿಮ್ಮದೇ ಬ್ಯಾಕ್ಗ್ರೌಂಡು. ನಾವು ಫಸ್ಟ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದಾಗ ಅದನ್ನೇ ಇಂಟ್ರಾಕ್ಟ್ ಮಾಡ್ತಿದ್ದೋ. ಅವರು ಒಂದು 35-40 ಜನ ವಿತ್ ಫೆಲೋಶಿಪ್ಪಲ್ಲಿ PhD ಮಾಡ್ತಿದ್ದಾರೆ ಬೇರೆ ಫಾರಿನ್ ಕಂಟ್ರೀಸಲ್ಲಿ. ಅಂದರೆ ನೀವು ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ, ನಾನು ಆಗ ಹೋಗ್ಬೋದು ಅಂತ. ವಿತೌಟ್ ಎನಿ ಇನ್ಫೀರಿಯಾರಿಟಿ ಕಾಂಪ್ಲೆಕ್ಸ್, ನೀನು ಹಾರ್ಡ್ ವರ್ಕ್ ಮಾಡಿದ್ರೆ ಚಾನ್ಸಸ್ ಆರ್ ದೇರ್. ಆ ಮೈಂಡ್ ಸೆಟ್ ಇಟ್ಕೊಂಡೇನೆ ಮತ್ತೆ ಯು ವಿಸಿಟ್ ಎನಿ ಸೆಂಟ್ರಲ್ ರಿಸರ್ಚ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್, CSIR ಯಾವುದೇ ನೋಡಿ, ನಮ್ಮ ಮೈಸೂರವರು, ಕರ್ನಾಟಕದವರು ಭಾಳ ಕಡಿಮೆ ಇರುತ್ತೆ. ಈಗ CFTR ಇಲ್ಲಿ ಅದಕ್ಕೇನೆ ನಾವು ಕನ್ನಡಿಗರನ್ನೇ ಕರೆಸಿದ್ದು. ಸುಹಾಸ್ ಇರ್ಬೋದು, ಬೆಟ್ಟದಯ್ಯ ಇರ್ಬೋದು, CFTR ಇಂದ ಯಾಕೆ ಕರ್ಸು ಅಂದರೆ, ನೀವು ಅಲ್ಲೋಗಿ ನೋಡಿದ್ರೆ ಗೊತ್ತಾಗುತ್ತೆ. ಮೋರ್ ದೆನ್ 80% ಬೇರೆ ಸ್ಟೇಟ್ ಅವ್ರು ಇರ್ತಾರೆ, ವೆಸ್ಟ್ ಬೆಂಗಾಲ್ ಅವ್ರು ಇರ್ತಾರೆ, ಬೇರೆ ಬೇರೆ ಕಡೆಯವ್ರು ಇರ್ತಾರೆ. ಅಂದರೆ ನೀವು ದಡ್ರು ಅಂತ ಅಲ್ಲ, ನಿಮ್ಮ ಎಫರ್ಟ್ ಇರಲ್ಲ, ನಿಮ್ಮ ಥಿಂಕಿಂಗ್ ಇರಲ್ಲ, ಆ ಡೈರೆಕ್ಷನಲ್ಲಿ ನಿಮ್ಮ ಯೋಚನೆನೇ ಇರೋದಿಲ್ಲ. ನಾನು CFTRಗೆ ಹೋಗ್ಬೋದು, ನಾನು ಸೆಂಟ್ರಲ್ ಕಾಫಿ ಬೋರ್ಡ್ಗೆ ಹೋಗ್ಬೋದು, ಬೇರೆ ರಿಸರ್ಚ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ಗೆ ಹೋಗ್ಬೋದು ಅನ್ನೋ ಡೈರೆಕ್ಷನಲ್ಲಿ ನೀವು ಯೋಚನೆ ಮಾಡ್ತಿರಲ್ಲ. ನಾವು ನಿಮ್ಮಲ್ಲಿ ನಾನು ಇಷ್ಟೇನೆ, ನಾನು BSc ಆಯಿತು, ಮ್ಯಾಕ್ಸಿಮಮ್ MSc ಮಾಡೋಣ, ಇಲ್ಲ ಅಂದರೆ ಬೇರೆ ಏನಾದ್ರು ಜಾಬ್ ಹುಡುಕೊಳ್ಳೋಣ ಅಂತ ಇರುತ್ತೆ. ಅಲ್ಲಿಗೆ ನೀವು ಸೀಮಿತ ಆಗೋದು ಬೇಡ. ಬ್ರೈಟಾಗಿ, ವೈಡಾಗಿ, ಬ್ರಾಡಾಗಿ ಯೋಚನೆ ಮಾಡಿ, ಭಾಳ ಅವಕಾಶ ಇದೆ. ಸೊ ಇನ್ ದಟ್ ಕಂಟೆಸ್ಟೇ ನಾವು ಎಲ್ಲ ಸ್ಪೀಕರ್ಸನ್ನು ಇನ್ವೈಟ್ ಮಾಡಿದ್ದು. ಈಗ ಲಾಸ್ಟಲ್ಲಿ ನೀವು ಟಾಕ್ ಕೇಳಿದ್ರಿ, ಈಗ ಜೆನ್ಜಿ ಹೇಳಿದ್ರು ಮೋಸ್ಟ್ಲಿ ಈಗಾಗಲೇ ಜೆನ್ಜಿ ಮುಗ್ದೋಗಿದೆ. ನಾನು ಮೊನ್ನೆ ಯಾರ್ದು ನಮ್ಮ ಹುಡುಗರು ಕೇಳ್ತಿದ್ದೆ, ಅದು ಆಗೋಯ್ತು ಸರ್, ಅದು ಹಳೇದು ನೆಕ್ಸ್ಟ್ ಲೆವೆಲ್ ಬಂದಿದೆ ಅಂತ. ಬಟ್ ಈಗ ನಿಮಗೆ ಪ್ಲೇಟ್ ಟು ಫುಡ್ ಕೇಳಿದ್ರಿ, ನೋಡಿದ್ರಿ, ಮೋಸ್ಟ್ಲಿ ಇನ್ ಫ್ಯೂಚರ್ ಹೇಗಿರುತ್ತೆ ಅನ್ನೋದು ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಎವ್ರಿಥಿಂಗ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ಕೆಮಿಸ್ಟ್ರಿ, ಎವ್ರಿಥಿಂಗ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ಇನ್ ಯುವರ್ ಹ್ಯಾಂಡ್. ನೀವು ಅದನ್ನು ಎನ್ಕ್ಯಾಶ್ ಮಾಡ್ಕೊಳ್ಳೋದು ನಿಮ್ಮ ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗಿದೆ. ಸೊ ಈ 6 ಜನ ಟಾಕು ಮತ್ತು ಬೆಳಿಗ್ಗೆ ಬಂದಿದ್ದವರು BSc, ಅವರು ಓದ್ತಿದ್ದವರು BSc ಹುಡುಗರೆ, 16 ಹುಡುಗರು ಬಂದಿದ್ದವರು BSc ಗ್ರಾಜ್ಯುಯೇಟ್ಸೇ. ನೀವು ಅವ್ರ ಜೊತೆ ಇಂಟ್ರ್ಯಾಕ್ಟ್ ಮಾಡಿ ಅನ್ನೋದಕ್ಕೋಸ್ಕರ ನಾವು ಭಾಳ ಶ್ರಮ ಪಟ್ಟದ್ದು. ಇಂಟ್ರ್ಯಾಕ್ಟ್ ಮಾಡಿ ಅವ್ರದ್ದು ಕೋರ್ಸ್ ಹೇಗಿದೆ, ನಮ್ಮ ಕೋರ್ಸ್ ಹೇಗಿದೆ, ಫ್ಯೂಚರ್ ಅವ್ರು ಏನು ಥಿಂಕ್ ಮಾಡ್ತಿದ್ದಾರೆ, ನಿಮ್ಮ ಥಿಂಕಿಂಗ್ ಹೇಗಿರಬೇಕು, ಹೌ ಟು ರೀಚ್ ಯುವರ್ ಟಾರ್ಗೆಟ್ ಅಥವಾ ನಿನ್ನ ಗೋಲ್ ಸಕ್ಸಸ್ ಮಾಡೋದು ಹೇಗೆ ಅನ್ನೋದ್ರ ಬಗ್ಗೆ ಇಂಟ್ರ್ಯಾಕ್ಟ್ ಮಾಡಲಿ ಅನ್ನೋದಕ್ಕೋಸ್ಕರ ಇದಿತ್ತು. ಬಟ್ ಏನಂದ್ರೆ ಬ್ಯಾಡ್ ಲಕ್ ಅಂದರೆ ಯಾರೋ ಒನ್ ಸೈಡೆಡ್ ಇಂಟ್ರ್ಯಾಕ್ಷನ್ ಆಯಿತು, ಆ ಸೈಡಿಗೆ ಬರಲಿಲ್ಲ. ಫ್ಯೂಚರಲ್ಲಿ ನೀವು ಯೋಚನೆ ಮಾಡ್ಕೊಂಡು ನೆಕ್ಸ್ಟ್ ಯಾವ್ದಾರು ಕಾನ್ಫರೆನ್ಸಿಗೆ ನಾವು ಕರೆದ್ರೆ ದಯಮಾಡಿ ಒಂದು 10 ಕ್ವಶನ್, ಸುಮ್ಮನೆ ಯಾವ್ದಾರು ಕ್ವಶನ್ ಆಗಲಿ, ಎನಿ ಕ್ವಶನ್ಸ್, ಸೊ ನೀವು ರೆಡಿ ಮಾಡ್ಕೊಂಡು ಬನ್ನಿ. ನಮ್ಗೆ ಕೇಳಿ, ನಮಗೆ ಗೊತ್ತಿದ್ರೆ ನಾವು ಹೇಳೋಣ, ಅಥವಾ ಬೇರೆ ಯಾರು ಎಕ್ಸ್ಪರ್ಟ್ ಇರ್ತಾರೆ ಗೊತ್ತಿರೋ ಹೇಳ್ತಾರೆ, ಅಥವಾ ಗೊತ್ತಾಗದೆ ಹೋದರೆ ಟ್ರೈ ಮಾಡ್ತಿರ್ತೀವಿ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಆನ್ಸರ್ ಮಾಡಲಿಕ್ಕೆ. ಸೊ ಆ ಥರ ಕ್ರಿಯೇಟಿವಿಟಿ ನಿಮ್ಮ ಮೈಂಡಲ್ಲಿ ಇರಬೇಕು. ಸೊ ವಿತೌಟ್ ಎನಿ ಎಜಿಟೇಷನ್ ಎಲ್ಲರೂ ಪಾರ್ಟಿಸಿಪೇಟ್ ಮಾಡಿ. ನೆಕ್ಸ್ಟು ನಿಮ್ಮ ಫ್ಯೂಚರು ಚೆನ್ನಾಗಿರುತ್ತೆ. ಐ ಥಿಂಕ್ ಈಗ ನಾನು ಬೆಳಗ್ಗೆ ಒಬ್ಬ ಪ್ಲೇಸ್ಮೆಂಟ್ ಆಫೀಸರ್, ದೊಡ್ಡ ಲೆವೆಲ್ ಪ್ಲೇಸ್ಮೆಂಟ್ ಆಫೀಸರ್ ಜೊತೆ ಇಂಟ್ರ್ಯಾಕ್ಷನ್ ಮಾಡ್ತಾ ಬರ್ತಿದ್ದೆ. ಇಲ್ಲ ಒಂದು ಫಂಕ್ಷನ್ಗೆ ಹೋಗಿದ್ದು, ಮಧ್ಯೆ ಇನ್ ಬಿಟ್ವೀನು ಅವ್ರು ಹೇಳ್ತಿದ್ರು, ಸರ್ ಈಗ ಇನ್ನು ಮುಂದೆ ಇರೋದು ಫ್ಯೂಚರ್ ಕೋರ್ಸಿಸ್, ಫ್ಯೂಚರ್ ಡಿಮ್ಯಾಂಡ್ ಈಸ್ ಸೈನ್ಸ್ ಓನ್ಲಿ ಸರ್. ಈಗ ನಮ್ಮಲ್ಲಿ ನಾನು ಕೇಳಿದೆ ಹೇಗಿದೆಯಪ್ಪ ಪ್ಲೇಸ್ಮೆಂಟು ಕಂಪ್ಯೂಟರ್ ಸೈನ್ಸ್ಗೆ, ಇನ್ಫಾರ್ಮೇಷನ್ ಸೈನ್ಸ್ಗೆ ಅಂತ. ಸರ್ ಅದಕ್ಕೆಲ್ಲ ಅವೆಲ್ಲ ಸ್ಟ್ಯಾಗ್ನೆಂಟ್ ಆಗಿದೆ. ನೆಕ್ಸ್ಟ್ ಕೋರ್ಸ್ಗಳು ಮಾಮೂಲಿ ಸೈನ್ಸ್ಗೆ ಭಾಳ ಡಿಮ್ಯಾಂಡ್ ಬರ್ತಾ ಇದೆ. ಬಟ್ ಹಾರ್ಡ್ ವರ್ಕ್ ಮಾಡಬೇಕು, ಕಾನ್ಸೆಪ್ಟ್ ಗೊತ್ತಿರಬೇಕು, ಕ್ರಿಯೇಟಿವ್ ಆಗಿ ಯೋಚನೆ ಮಾಡಬೇಕು. ಅದರಿಂದ ಈ ಥರದ ಕೋರ್ಸ್ ನೀವು ಮಾಡಿರೋದು ಎಕ್ಸಲೆಂಟ್ ಇದೆ. ಬಟ್ ಅದನ್ನು ನಿಮ್ಮ ಮಕ್ಕಳಿಗೆ ಎನ್ಕ್ಯಾಶ್ ಮಾಡ್ಕೊಳ್ಳೋಕೆ ಹೇಳಿ ಸರ್. ಫ್ಯೂಚರ್ ನೆಕ್ಸ್ಟ್ 3-4 ಇಯರ್ಸ್ ಆದರೆ ಭಾಳ ಡಿಮ್ಯಾಂಡ್ ಇರುತ್ತೆ. ನಾವು ಮಾತ್ರ ಇಲ್ಲಿ ಥಿಂಕ್ ಮಾಡೋದು ಮೆಡಿಕಲ್ ಮಾಡಿದ್ರೆ, ಇಂಜಿನಿಯರಿಂಗ್ ಮಾಡಿದ್ರೆ ಸ್ಕೋಪ್ ಇದೆ ಅಂತ. ದಟ್ ಈಸ್ ರಾಂಗ್. ನೀವು ಯಾವುದೇ ಬೇರೆ ಕಂಟ್ರಿಗೆ ಎಂಟ್ರಿ ಆದ ತಕ್ಷಣ ನೀನು ಮಾಸ್ಟರ್ ಡಿಗ್ರಿ ಇದ್ದರೆ, ಯು ಆರ್ ಎಕ್ಸ್ಪರ್ಟ್ ಇನ್ ಸೈನ್ಸ್, ದೇ ಗಿವ್ ಫಸ್ಟ್ ಪ್ರಿಫರೆನ್ಸ್ ಫಾರ್ ಯು ಪೀಪಲ್. ನಿಮ್ಮ ನಾಲೆಡ್ಜ್ಗೆ ವ್ಯಾಲ್ಯೂ ಕೊಡ್ತಾರೆ, ನಿಮ್ಮ ಕೋರ್ಸ್ಗೆ ಗೌರವ ಕೊಡ್ತಾರೆ. ನೀವು ಅದನ್ನು ಎನ್ಕ್ಯಾಶ್ ಮಾಡ್ಕೋಬೇಕು. ಇನ್ ದಿಸ್ ಫ್ಯೂ ವರ್ಡ್ ಎಲ್ಲರೂ ಇನ್ವಾಲ್ವ್ ಆಗಿದ್ದೀರಾ, ಬೆಳಗ್ಗೆಯಿಂದ ಕೇಳಿದ್ದೀರಾ. ಸೊ ಏನೇನು ಗೈನ್ ಆಗಿದೆ ಅದನ್ನು ನಿಮ್ಮ ಮನಸ್ಸಲ್ಲಿ ಇಟ್ಕೊಂಡು ಅಳವಡಿಸ್ಕೊಳ್ಳಿ. ಏನಾರು ತೊಂದರೆ ಆಗಿದ್ರೆ ಅದನ್ನು ಇಲ್ಲೇ ಮರೆತು ಮುಂದೆ ಹೋಗಿ. ಒಳ್ಳೆಯದಾಗಲಿ. ಗುಡ್ ಲಕ್.
Thank you, sir. We now move to an important segment of this valedictory ceremony, the best poster presentation award. And before closing, I request the award winners to come and collect the certificates. Krupanjali Devi, CSIR Sri Mangalore. Harshita Vayyam, Jesus College for Women, University of Mysore. Priyanka, Jesus STU, Mysuru. Shivakumar BP, sir, Jesus STU, Mysuru. Congratulations to all the award winners. As we conclude the international conference, we carry forward valuable knowledge, new collaborations, and renewed inspiration. On behalf of the Department of Chemistry, I sincerely thank all the dignitaries, speakers, participants, and our organizers for making this conference a grand success. Thank you all. Wishing you a safe journey and continued success in your academic endeavors. High tea has been arranged for all. I request all the participants to join us for that. Thank you all.